It gives me immense pleasure to welcome you all to this 5-day faculty development program on emerging trends and innovations in post harvest technology and management towards sustainable food systems. Today's session title is Crop Improvement for Minimizing Post Harvest Losses and Improving Nutritional Quality of Horticultural Crops. As we all know that horticulture plays a pivotal role in ensuring food and nutritional security, enhancing farm income and supporting sustainable agricultural development. Fruits, vegetables, flowers and plantation crops are not only high valuable commodities but also vital source of vitamins, minerals, antioxidants and other bioactive compounds essential for human health. However, despite significant advancement in production technologies, a substantial proportion of horticulture produce is lost after harvesting due to physiological deterioration, mechanical damage, microbial infection, and inadequate storage and transport systems. The post harvest losses directly affect farmers' income, market stability, and national food security. Conventional and molecular breeding strategies are hence essential for improving post harvest quality, focusing on extending shelf life, enhancing firmness, reducing water loss, and increasing resistance to pests and pathogens. Conventional methods like hybridization and selection are often combined with advanced molecular techniques such as CRISPR-Cas9, marker assisted selection, and omics technologies to achieve these goals with higher precision and speed. Breakthrough in genome editing via CRISPR-Cas now allows scientists to precisely target senescence-related genes, such as those controlling ethylene biosynthesis, effectively extending the shelf life of highly perishable crops like tomatoes, bananas, and avocados. Simultaneously, nutritional biofortification has advanced beyond traditional breeding, transgenics, and editing platforms are being used to enhance health promoting compounds, including zeaxanthin rich tomatoes, beta carotene enriched eggplants, and high iron leaf vegetables. This particular 5-day session of FDP is designed to provide a comprehensive understanding of the genetic, physiological, and molecular basis of post harvest traits and nutritional quality. In this context, it's my great honor to introduce our distinguished speaker, Dr. Sashi Bhushan Tripathi sir, Professor, Terry School of Advanced Studies, New Delhi. Dr. Sashi Bhushan Tripathi sir is a professor in Department of Biotechnology at Terry School of Advanced Studies, New Delhi. With over 20 years of experience, he is a leading researcher in molecular breeding and germplasm characterization. He has used various molecular markers in several plant species for analysis of genetic diversity and population structure. Since 2014, he has been using genotyping by sequencing markers in Jatropha, okra, chili, cattle, poultry for germplasm characterization and identification of QTLs for important traits. He has been an active researcher in several DBT funded network projects on germplasm characterization such as tea, bamboo, agarwood, jatropha, and sea buckthorn. His project on development of double low varieties in Brassica juncea has been awarded for Best Innovation Award in Agricultural Biotechnology by BIRAC, Government of India. Currently, he is a part of DBT funded network project on banana genetic diversity. In addition, he is also working on genetic improvement of Bhut Jolakia, a chili landrace of Assam. The funding agencies, there are a whole list of the projects that he has been associated with, and the funding agencies are DBT, DST, BIRAC, Indo Austrian DST, Indo Russian Project, Swedish International Development Agency. So basically, Sir is going to talk on the topic application of DNA markers in genetic improvement for nutritional quality and post harvest management. We welcome you, sir, and it's over to you.
Thank you so much.
Welcome, Dr. Shashi Bhushan. Thank you,
Yes, yes.
thank you.
So am I audible?
I'll join little later.
But am I
Yes, you
audible?
are. Thank you.
Yes, sir, you are audible.
Okay, okay. So thank you so much, Dr. Verma, for
Yes, sir.
this great introduction, and special thanks to Dr. Nutan Kaushik, my colleague at Terry, for giving me opportunity to share with you the uh, our experience in the field of this molecular markers. So give me just few minutes to upload my the screen. So is my screen visible to you?
Yes, sir. Yes, sir, it's visible.
Yeah. Okay, so I think now the screen is visible very well.
Yes, sir.
Yeah, so this lecture on application of DNA markers in genetic improvement for nutritional quality and post-harvest management. Uh, I'm really grateful to all the organizers. Now, just to give you the introduction, and I believe the audience is a mix of faculty, students, researchers. So I will keep my this level of the talk, trying to balance with all the stakeholders. Now we know that genetic improvement it is like systematic process of enhancing desirable traits, and every time you'll hear new varieties are developed and released. So recently, in the beginning of this year, we remember our Minister of Agriculture, Mr. Shivraj Singh Chauhan. He released several varieties, 184 varieties, which included varieties of cereals, pulses, and then several fruits also, oil seeds. So this is a always a continuous process, the development of new varieties. And every time a breeder wants to develop a variety, the primary trait for improvement is yield, because yield is something which also helps the farmer to get maximum this remuneration. Because most of the time we pay based on generally the weight, and generally we are not very much focused, at least the farmer is not able to sell the crop based on the quality. So that is why yield is definitely the primary trait. But with the self sufficiency in grain production and crop production in India, uh, of late there has been a shift towards nutritional quality also. So people want good quality, and also with the new regime of this climate change, this climate resilience and post harvest management has also become one important center of this uh, objective in the improvement. And lately we have recognized the issues such as hidden hunger and food safety. So we talk about sustainable development goals, and SDG 2 is about removing all the hunger by 2030. But at the same time, there is this hidden hunger which is about the malnutrition. So SDG 2.2 talks about malnutrition, and there has been a means now we are trying to include nutritional quality also in the objective of genetic development. Now, why we need this nutritional improvement? Because there has been several reports that there has been somehow uh, basically in the more poverty, people who are below the poverty, they have deficiency in different micronutrients, for example iron, zinc, vitamin A. These are the three major ones. So there has been always an intent to increase the content of these things in our regular crops. So you must have heard about iron and zinc. Means varieties with high iron and high zinc in case of millets. Also, vitamin A has been a center of study. So, golden rice. This was something which was targeted to meet this problem, solve this problem. And again, there is a segment who likes health value of the foods. For example, tomatoes of different colors, basically the which have high lycopene and high anthocyanin, also vitamin C. So people would like to pay for that. There is interest in functional bioactives, for example, polyphenols and omega-3 fatty acids, dietary fibers. So increasing these things in the product is always going to give a premium price. Uh, Also, there has been interest in having tastier food. For example, in case of soybean, there is a off flavor, especially in the products such as tofu and soya milk, and people have tried to solve that by reducing the lipoxygenase activity, also reducing the common anti-nutritional compounds, for example, phytic acid. Phytic acid, it is something which binds to phosphate and reduces the bioavailability of phosphorus, lectins, and protease inhibitors, which create problem in digestion of food. So, reducing these compounds in legumes and wheat, this has been already targeted. In case of post-harvest management, we know that a lot of product is lost. Just in the introductory part, Dr. Burma talked about this shelf life, and uh, we know that a lot of product it gets lost because either there is a problem in the storage or transportation, and therefore there are traits, for example, delayed ripening or improved firmness. These may be of interest um, because they will lead to less losses in during the post harvest. Also, there are problems like uh, there are issues such as anthracnose in chili or tomato or brinjal, which leads to getting the product which will be of not selling quality. In quinoa also we have seen this brown rot, which affects a lot of fruits and that creates means losses. So all these issues they can be solved by different means there can be different strategies. For example, one can increase the nutrient content, for example, iron, zinc, and these things, just by having a better agronomy, by providing those things as fertilizers, and that is one way. But as a geneticist, we are having this option of creating new varieties which have these traits, which will help either in having good quality or having less post-harvest losses. So I shall be mainly talking about the interventions which the geneticists or molecular biologists can do because agronomy and uh, having a very good logistics, a chain, a cold chain, that does not come into purview of us the biotechnologists. So if a breeder or a biotechnologist wants to have to do genetic improvement, these are the three approaches which are available to the person. So the first one, which we call breeding based approach, this is the conventional one. This is the traditional one can say, and most of the varieties of today they have been developed using this approach. So whatever we are eating in everyday life, most of it has come through this approach. In fact, in India, nothing which we are eating has come to through any other approach so far, because due to various reasons, the this has not been permitted. Now, this breeding-based approach has its own limitation. The main limitation is that for every improvement, we need a donor. So if you want to have a combination of traits, those traits should be there in the parental lines. We cannot generate a trait which is not there in any of the parental lines. And again, the traits should be available within the species gene pool. The species gene pool means those individuals which can be crossed. If there is a trait which has to be improved in tomato, we have to find that trait in tomato only. If that trait is there in potato or say brinjal, we cannot bring that from there. So this will not be able to break the species boundary. So the trait has to be within the species gene pool. But then there are these two new approaches, the transgenic approach that requires only the knowledge of the pathway and target genes. If we know the gene which we want to have in the product, and with the available technologies, generally it is possible to develop a means a new product with the required traits. Um, so this does not have the problem of species boundary. It can a gene from bacteria can be inserted into a tomato, and a gene from any other species can be transferred across the species boundaries. Uh, However, there has been a lot of resistance from the different sections of stakeholders, and somehow in India at present we have only one crop where this transgenic approach has been utilized, and that has been really successful. The BT cotton, we all know, this is a great example. But for all other crops, they are in various stages of development or regulatory clearance. So somehow the regulatory clearance is not forthcoming that easily. The third technology, which is relatively newer one, the gene editing, and this has given us an opportunity. So the basic criticism in case of the second one, the transgenic approach, is that the product contains uh, DNA or genes from a non-aligned species. So a bacterial gene may be present in tomato. So that is why people say maybe we don't know how that is going to affect those who are going to take that. Now, in case of gene editing, that problem is somehow solved, because here we have the option of very precise conversion or very precise editing of the uh, select portions of the genome, maybe a single nucleotide or a few nucleotides, so that we get a particular trait. But that again requires a very well understood target genetic sequence for the trait. A lot of information is required for that, and I hope this technology, gene editing, will take up in coming years. I work basically in the domain of the first group of technology, that is basically conventional breeding based approach, and this is actually a very you can think of a very simple process, like you select the parental lines which have the desirable traits which we want in the variety final product, and then we make a cross, and using the principles of genetics, we try to create a new combination. And for that, we have to do this cycles of generation advancement and selection. And this selection and generation advancement can go and it can be taken forward in different ways, in different means how many cycles this will happen, whether the generation advancement will happen through selfing or open pollination or back crossing. So all these things may be different, but generally the process is like one has to create a population where you can make selection, and uh, the breeders try to select the best one from the population, and then they evaluate it, multiply it, and then it is the same thing. The product is released as a variety. Now, in this whole process, there is the issue like this whole process is very lengthy process. It is not that swift. For example, the major portion, major time is taken in the second step, that is generation, advancement, and selection, and it may take several generations. Now, based on whether we are working on a crop which can complete its life cycle in 4 months or 6 months or 1 year or 5 year, means seed to seed cycle, this time can be different. And in general, in any annual crop, like you can take the example of rice or wheat, it takes at least 10 to 12 years to develop a new variety from scratch. Now, the question is, can we speed up this process? Means, can we make this time shorter than this? Because if we can do that, there are means benefits to if you are a seed company, and if you are able to release a variety 2 years earlier, you have a market advantage. So that way, uh, every company or every breeder would like to have this development of new variety at the earliest. Now there are these different options are there. So the first one is, uh, if we can take multiple generations per year. So suppose we are talking about an annual crop and it takes 4 months from seed to seed, for example wheat, and if we can take multiple generations, then this time can be reduced. But that is a logistic approach. Like for that you need systems, you may need infrastructure. For example, this shuttle breeding, which is very common in case of wheat and rice, where people uh, put the crop in one place in one season and then take the next generation to another place where um, the environment is conducive for growing that. Again, this new, relatively new approach called speed breeding. This can this can do this. Uh, this can this can complete at least 4 to 5 generations in case of wheat in 1 year. So, which means 2 months seed to seed cycle. So these things have definitely become of very great use, and people are getting help from these approaches. But at the same time, if we can do, if you can increase the accuracy of selection, then that will be an additional thing. And many times, this selection is a major problem. One is selection accuracy, that when we are selecting based on phenotype, many times the phenotype is affected by environment, and we may not be selecting the right individual. Maybe that has got complicated by environment, so we are not selecting the correct one. Also, if we can select the individual at an early stage, that will be also great, because in many cases this will shorten the this breeding cycle. And to select the individuals at early stage, there are two major approaches, two very commonly used approaches. Marker assisted breeding, which basically looks at few number of genes, few number of loci at a time, and genomic selection, which can tackle a large number of loci. So the approaches are slightly different. And the principle of this marker assisted breeding and genomic selection, they are different, but both the approaches people have started, and using these, people have demonstrated that actually the time of this release of variety can be shortened very effectively. So here, um, as a case study, I am giving you this example of a project which we did uh, sometime back, 2011 to 2015, and this was the project which was funded by BIREC, that biotechnology industry partnership program, BIP program. And in this, we collaborated with a seed company called Nirmal Seeds at Jalgaon, Maharashtra. So, Madam Kaushik was the PI in this, and I was the co-PI. This was one project where we worked jointly. And to give you some background of this, what we wanted to do in this project was that we all know this Brassica gentiana, Indian mustard, because this is a major oilseed crop in India. Means it is one of the most utilized type of oil. Uh, but although it is very commonly used, there have been reports that this seeds they contain two very important major anti-nutritional compounds. One is glucosinolates, which are nitrogen sulfur compounds, and they are basically they create problems in the digestion. So basically, this glucosinolates they are present in the seed meal, and we know that most of the seed meal or oil cake we use as cattle feed. So if cattle are fed with this uh, oil meal, there will be this problem in digestion. So this is one issue. The second is it contains a monounsaturated fatty acid, long chain monounsaturated fatty acid called erucic acid, and erucic acid is present only in Indian varieties of Brassica gentiana. So this is a major fatty acid. Almost 40 to 45% of total fatty acid profile composition is erucic acid, and this has been linked to coronary heart diseases. So now with these anti-nutritional compounds, one would definitely like to reduce these components in the variety. So all varieties which are available in of Indian mustard, they have these glucosinolates and erucic acid. So you can see the products like mustard seeds, and from that you have these two major uh, products. The oil contains the almost 45% erucic acid, and oil cake it contains glucosinolates. So glucosinolates they are present almost 150 micromole per gram of the seed cake. So it is very high. Now we know there are products, especially from other countries, and they also belong to the same uh, genus Brassica, but it is a different species called Brassica napus. And this species, this is mainly grown in cold countries like Canada, and there is an oil called canola oil. In fact, oil from Canada, so that is canola, and this is nothing but mustard oil only, but it has very less glucosinolates, almost free from glucosinolates and erucic acid. Now, one way could be that we grow this Brassica napus varieties here and get the canola quality oil. But the issue with this species is that it requires very chilling conditions, which is not present in India. So maybe we can grow only in the hilly places, some places, but overall, the main growing area of Brassica gentiana is the central India and central and northern India. So there we cannot have that very high chilling temperatures. So our varieties, our Brassica gentiana varieties, they are adapted to this condition. Now the other option can be that we convert these our Brassica gentiana varieties into a product, into a variety which has very low glucosinolate and erucic acid content. So as a standard, if the mustard variety has less than 30 micromole per gram glucosinolate and less than 2% erucic acid in the oil, it is called canola type. All people also call low, double low mustard. So this is a quality standard. So the question is, can we convert our varieties, our Indian mustard varieties into canola type? So as a geneticist, it is a very straightforward problem. It is like just take the genes from Brassica nepus and convert means bring it to Brassica gensea. So in a theoretical level, it is very simple, and this is what we did. So we transferred the low alleles. So we call them low alleles because those alleles of this genes, they create low levels of glucosinolates and hydroxylic acid. And this is what we did in our project. Now, any breeder or any person who has who had who had studied genetics, the person knows that if we want to transfer just few genes, one or two or three genes to another uh, background, the backcross breeding is the approach. So backcross breeding actually it is the approach which is used in a situation when we want to uh, just transfer one or few genes to an existing variety. So the existing variety must be very good, but it is lacking those one or two genes, and those genes one can bring from another line, another type. So there are this so-called donor line, which can give the trait, those one or two important traits, and a recipient line, which is actually an existing variety, which is already being grown. So it is a uh, kind of already is in use. Only you want to convert it. So we have in this backcross breeding this three important things which we have to achieve. The achievement should be like, first we should be able to select for the target gene or target trait. So this is what we call foreground selection. At the same time, we should be able to recover the genome of the existing variety, which is called recurrent parent genome. So, and this is done by a method which we call background selection. So foreground selection and background selection both have to be achieved. Another issue which is means faced when we want to do this backcross breeding is the chromosome on which the gene of interest is there. There, there will be a bigger segment of the donor variety, and the donor line that those segments may have some you can say negative genes or maybe not desirable genes. So we want to get rid of that also. That is what we call genetic drag. So I just give you this figure, which, and I'm just trying to have slightly some theoretical things also. So this is the variety which we call recurrent parent, which is a good variety otherwise, but it has one or two genes which are not there. The donor parent, it is a variety which is not that good variety, uh, but it has this gene which is in which we are interested. If this was a good variety, then we could have directly used it as a variety. There was no need of this backcross breeding. But we don't want other parts of this donor parent genome. We just want this small part of the donor parent genome so that we have most of the genes from the recurrent parent genome and only this small part from the donor parent genome. So this will be our target variety. This is what we want to make. So to do this, the process is the backcross scheme or backcross breeding, where you cross the donor and recurrent parent, and then we know that when we make a cross, such a cross, the F1 hybrid will have 50-50% of the genomes of the both parents. Now we want to reduce the genome of this donor parent. So what we do, we again make a cross with the same recurrent parent, which we call back cross. So when we make a back cross with this recurrent parent genome with and F1 hybrid, we get what we call a back cross 1 population. And from back cross 1 individuals, again we cross with the same recurrent parent and we get the second back cross. So this way we can continue several generations. This is what I'm talking about, the conventional method. So when we don't have any support from markers. So in that case, we can go like this, and after several generations, say 6 to 8 back crosses, you will have most of the genome from the recurrent parent genome and only a very small portion of um, donor parent genome. And in throughout this process, you have to ensure that you have not lost the gene which you want from this donor parent genome, because every time the genome of donor parent is getting lost, we should not lose that part which is important for the trait. Okay, so the marker assisted back cross breeding, uh, it is like a support to the breeders. So if a breeder wants to go ahead with this system, it is fine. But the issue with this system is that it takes a lot of time, especially when the trait is recessive, because we know that if there is a trait which is recessive, and that is important, when we make a cross with the dominant, the F1 hybrid will have a heterozygote, but the trait, recessive trait will not be visible. This is what we have studied in our basic genetics. So F1 does not show the recessive genes. Now, this plant we have crossed again with the high, and in the first back cross, what will happen? You get these two kinds of genotypes. One, the homozygous one, which is anyway going to be dominant, and this heterozygote, which also show the dominant trait, because it is again heterozygote. It will not show the recessive gene. Now we have to make a selection from these two. Now we have to somehow um, differentiate between these two. If we go by the trait, or what we call phenotyping, so if we take the seeds of these two plants, they all will be showing high glucosinolate or high erucic acid. So we cannot make a distinction between these two. But now there are this so-called marker-assisted breeding, where if we have developed a marker for this trait and for these alleles, then we can simply use those markers as a proxy. Like we can just see the marker and we know what we are actually going to get. For example, in this case, if you see the homozygote and heterozygote, although it will not be distinguishable by using this phenotyping approach, but the genotypes you can see, they are very clearly different. This is showing two bands, this is showing single band. So we can tell, okay, this is the heterozygote and this is what we want in the next generation. So this way, it is very easy to find out which of these two we want, and the distinguishing the heterozygote from homozygote is possible by using this marker, the different markers. Uh, Generally, if we don't have a marker support, what people do, they they use both of the lines, both of the plants for the next round of backcross, and then they use the genetic analysis, and then they try to find out, okay, we expect segregation here and no segregation in this case. So from that genetic analysis, they confirm that, okay, this is the heterozygote and this is the homozygote, but it will take one full generation, which means if you want to go for 6 generations, it will take 12 generations if you are using a conventional approach. And if you are using a marker assisted approach, you can complete it in 6 generations. So the generations will be halved, and that is a great saving in time. So the selection, how we do the selection is, in the previous slide I showed you that the F1 has 75% of the recurrent parent genome and 25% of the donor parent genome. But this is not true for all plants. It is true for only the average population. So if we have all the plants, we see all the plants, so there will be plants which have where only 60-65% of the recurrent parent genome, and there will be plants which will have as high as 90-92% of the recurrent parent genome. This is what we achieve through background selection. So our objective is from these different plants to find this group of plants which are very small in frequency, because you can see the frequency of these tails are going to be very small. So out of say 100 plants or 200 plants, you may get only 5 or 10 plants which have this very small, sorry, very high recurrent parent genome. So to find this, we use the so-called background selection or background markers. And background markers basically they target or they try to see the alleles of the recurrent parent genome. So we are interested in the alleles of recurrent parent genome. So background markers they basically see the alleles of the recurrent parent genome. And from this genotyping, by using this so-called genotyping, we can create what we call graphical genotypes. And these graphical genotypes are something like we are painting the chromosomes and showing, okay, this part of the chromosome is homozygous and this is heterozygous. Which part is homozygous and which part is heterozygous? We can just paint, we can tell by the color. And suppose if you are interested only in this portion, so we'll select this individual and not this individual, because this individual has a bigger chunk of the this chromosome. We want a very small portion of this chromosome. So this way, you can select out of so many individuals, which individual is of use, which one individual is having the least amount of the donor parent genome and highest amount of the recurrent parent genome. Now, this marker assisted breeding approach requires availability of markers, and that is done by a separate means research. Like one has to invest time and research in finding the markers linked to the trait. And for glucosinolate, the research group of Dr. Akshay Pradhan at the University of South Delhi South Campus, they had found that four loci or four genes situated on different chromosomes, they are important, and they actually work in a context dependent manner, which means if only one of the gene is there, then it will have 137 micromole per gram of this glucosinolate. But if we have both the genes together, A2 and A3, we can achieve 25 micromole per gram, which means if we just use these two genes, we achieve the canola quality. Canola quality is 30 micromole and lower. So this is lower than that. So this is what we used. We used these two markers for selecting the uh, individuals so that we can get the low alleles of these two. For that, these are the markers for the glucosinolate content. So you can see these are very clear markers, codominant markers. So they can very easily tell you whether it is a homozygote or heterozygote. So both you have the low glucosinolate allele, and this is the high glucosinolate allele. So we are interested in this one. Okay. For the second marker also, we are interested in this upper one. So for the other trait, the erucic acid, again there are two major genes, which are important and which regulate the concentration of or the composition of this erucic acid. So these two genes, when they are present in homozygous dominant condition or wild type homozygous, they will have the highest amount of erucic acid. But if all of these are in the form of homozygous recessive, then you can achieve this less than 2%. So this again have additive gene action, like if one is present in dominant another is present in recessive, you will get a middle one. So it will be somewhere around 20-25%. So we use this, means we converted this information into a usable markers, which you can see here. So the sequence, we sequenced the different alleles, and from those we designed an assay method which can distinguish between the low allele and high allele, both in a polyacrylamide gel system as well as in a agarose gel system. So the lower allele, you can see the it is visible even in the agarose gel system, and use these markers for selecting for the low erucic acid. So these two sets of markers, the for this low erucic acid as well as the low glucosinolate marker, these we used for marker assisted backcross selection. Now in this case, we used a donor line called TERI4, which was developed again. Dr. Nutan Kaushik's group had developed it, but this line had the good trait is that it had low erucic acid and low glucosinolate, but the issue was that it has very small seed grain, very small pod size, and it was not. I mean, agronomically, it is not good. So we wanted to use this as the donor line. The second line, the um, recipient line, was called NM100, actually NML100, and this was a line which was commercial line by which was being used by the company Nirmal Seeds. So this was a commercial line, a superior line, and we wanted to just uh, convert this into a double low line. So the donor line and the TERI4 and this recipient line is NM100. And if you see here, the erucic acid content in TERI4, it is non-detected. In the commercial line, it is very high, 45%. And glucosinolates also here, it is very less, 2.94 micromole per gram, and here it is high, 93 micromole per gram. So with this information, we use this breeding scheme, and this scheme is, I have already shown you that cross the donor line and recipient line, F1 again crossed with the recipient line, and that way every back means round of back cross we are screening the individuals for the low alleles, and generally it takes 6 to 8 generations to achieve 95% or some 98% of recurrent parent genome, but we achieved in just 2 back crosses. In the first back cross itself, we achieved 91%. This was because we used this background selection, which I showed you here. So in the first back cross itself, we selected these individuals which had somewhere around 91% of the recurrent parent genome, and then we again crossed that for the second round, the back cross 2, and then we got a very high amount, almost 95% of the recurrent parent genome. And then the anyway one has to do the selfing because you want to make the plants homozygous for the recessive trait, and from the recessive individuals we selected again the homozygous lines with double alleles. So some pictures you can see in the which are related to this project. So you grow the lines and then you make crosses. Um, After that, you do isolation of DNA from these different. Every individual has to be tagged, and then you isolate DNA and do the genotyping. From the genotyping, you can easily select. Okay, these are the individuals which are of interest, the heterozygous ones. These are homozygous. These are not means desirable. We don't want them. So these individuals one can easily select, and then we go for the other with other markers. So all the markers we use one after other. So this is screening for the um, this erucic acid alleles, and we know that okay, these are the individuals which are good for us. So when you start with say 400, 500 individuals, you get few individuals, say some 30 individuals or so, and those you use for background selection. For background selection, we used a marker system commonly called AFLP, Amplified Fragment Length Polymorphism. And there you can easily use these markers which are polymorphic, and based on this data, one can find out what is the similarity between each of them. In fact, we are interested in the similarity with the parent, the recurrent parent, the NML100. So we found that these two lines, Becross 1, 535 and 589, they had very high similarity with NML 100, almost 91%. And then we used them for the second round of Becross. So from that, we did again, you do the same round of DNA isolation and screening. So finally, we achieved the so-called our target variety, and the target variety not only has to be means selected through markers, also it has to be seen in the field. So for the agronomy, for example, here if you see the donor line and the donor line is this one, and the recipient line, the new line has more similarity with this recipient line, because we want this type of plants only, not this type. Here you can see the leaves are serrate. Here the leaves are dented. So this also has dented leaves. Same way you have, if you see the flowering time, so the two lines, NML 100 and the new line, both have the same flowering time. The Terry 4 is slightly delayed. It flowers slightly delayed. So this shows the that selection has happened very well. Even at the pod level, you can see the pod size of the Terry 4 donor line is very small. The animal 100, the recipient parent, it is bigger, and the new line also has almost same size of animal 100. This the pods. So anyway, in case of mustard, we always want bold seeds and bold pods. So that way, this is very clearly shown that we have achieved this recipient parent genome in the new line. When we tested the erucic acid and glucosinolates in the line, you can see the erucic acid almost not detected, and glucosinolates in two of the lines, it was 20 micromole per gram, and in two of the lines it was 30 micromole per gram. So one can easily release this, and in fact the company, they use this in their commercialization. So in fact this was the first variety of double low canola type in case of Brassica jonesii. After this, in 2017-18, PUSA, the ICR, also has come up with double low and single low lines like PM 29 to 30, PM 30, and PM 31. But this was the first variety. And this study also demonstrates the successful implementation of marker assisted backcrossing. Uh, Now, here if you see, we have used EFLP markers which were available at that time, but now we are using the next generation sequencing based genotyping platforms, for example, DDRED and genotyping by sequencing. So these platforms generate much bigger data with very high resolution, and so that provides us a very improved means accuracy of selection. So the power of marker state selection increases with those. In fact, these new next generation sequencing platforms, they have made the possibility of genomic selection. So I talked about genomic selection. Genomic selection requires much high resolution of marker data. So this is the kind of platforms which are providing those data. So I think this is the last slide, and mm, uh, just to show this picture, if you can see, this was the Bayrak Award in 2015, and Madam Dr. Nutan Kaushik, you can see here. At that time, the minister, our, our Minister of Science and Technology, Dr. Harshvardhan, was also there. So this is the picture. 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 So thank you so much. If you have any questions, I am available. So acknowledgements to the BIRAC for the funding, the company Nirmal Seeds for collaborating with us because this was very important. One cannot demonstrate this without this collaboration. Also, Dr. Professor Akshay Pradhan for sharing the markers. In fact, I had a earlier project with him on the same this for development of these markers, and also our uh, parent organization TERI, the Energy and Resources Institute, for allowing us to work on this subject. Thank you so much.
Thank you, sir, for sharing your valuable insights and experience with us. This session is open for question and discussion now. If anyone of you have any doubts or queries, you can post in the chats or can raise hands to directly talk to our expert. Is there any question? Audience, if you want to ask any question, you can put it in chat or you can raise your hands also. Okay, so by that time, I have some questions to ask to you.
Sure.
Because your this presentation was on markers, so I also have a very tiny experience to work with RAPD and SSR during my PhD tenure. But in your this PPT, you have discussed about speed breeding and this shuttle breeding. So basically, one is conducted at a particular controlled environment, and one is conducted from one place to another. Like you cannot conduct it at the same place. So, is there any example or any variety that has been released in particularly in terms of horticulture department through these methods?
Mm.
Or like, I just want to know the
Actually,
development whether we are taking it into action like a practical way we are incorporating this or not.
uh, now when we talk about horticultural crops, then this includes so many things. So it can include annuals like the vegetables, mainly the vegetables, tomato or chilies and beans, such crops. Also, it can also include fruits, which are perennials. Now, this speed breeding and this cattle breeding has been mainly used for cereals, where you just want to advance the generation by selfing. In most of these breeding of these horticultural crops, pure lines are not very common. Means, in fact, the even in case of annual crops like tomato or chilies, if you see, almost 95% of the market is for the hybrid seeds. The companies don't want pure lines. Maybe some only the universities, the agricultural universities, state universities, and central universities, they develop some of the pure lines, but in general, people are interested in F1 hybrids because that gives them control over the seed. It cannot be pirated that easily, the F1 hybrid seed, and everybody, the farmer has to come every year, every season. In case, so I don't find actually any example of shuttle breeding or speed breeding because this is not possible. If you want to uh, generate a horticultural crop, for example, fruits, it will be difficult for growing that in that way. They are perennials. So, and also in horticultural crops, the method of breeding is different. So, mainly they use an approach which we you can call it hybridization, but it is not the same hybridization. When we have make hybrids, we cross pure lines. So the two pure lines they are crossed, and we get the hybrids F1 hybrid. This is not the case in like fruit crops or anything. There we don't do this type of crossing because we don't have the option of getting these pure lines. Um, Mango and this banana. In case of banana, again there is a different issue. In case of citrus, so there people make crosses whatever is there, and then it will be like a full sheep population, like a population which we call full sheep population, and from that people select. So it is much similar to what people who are working on animal genetics they are doing. The same approach people who are working on and perennial crops like perennial horticultural crops or trees. People use that approach only.
I'll take few questions from the audience. There is one Samad Raza, a scientific officer in horticulture from Pakistan, and his question is: Are there any methods other than breeding that can be used to biofortify mango crops, given that mango is not a cereal and is not available year round? Additionally, can the simple application of zinc and iron biofortify the fruit?
So I'm not the expert in this field, so it will be difficult to comment on that.
Mm,
But definitely, what I can understand, there must be products. You can say the processing.
mm,
Processing can definitely increase the content of some of these things. For example, we have the example of biofortification in milk, biofortification in different so-called infant formulas. So similar processing may be possible. So the products which are made from mango that can be made biofortified. Uh, Per se, the fruits of a tree, I don't think that is so easy because
Yeah, without breeding, I mean, you can do one thing that you can just coat the fruit with some and use some
yes,
like edible coating and
edible
add
coating
some.
or the pulp
Uh,
you can process and there you can mix whatever you want. So people must be doing that very commonly.
yes, sir. And the next question is from Dr. Charugupta, ma'am, and she's asking, I want to know that we have read that glucosinolates have nutraceutical importance like antioxidant, anti-cancer activity, etc. Then why there was a need to reduce them in Brassica species, as you showed in one of your slides?
Hmm. So it is true. It is true that glucosinolates they have means health values. The thing is, the issue lies in the concentration or the amount. So if uh, we are consuming this mustard seeds in different forms uh, in our daily life, I don't think that is going to have any issue because there we are taking very small amount of that. But I was talking about the animal feed or cattle feed where the that oil cake, is given in very large amounts, and it is a common fact. Means, in fact, personally, I have also seen that the cattle, if you feed lot of this oil cake from mustard, they will have a problem, digestion problem. So it is all about the amount. In lower amounts, definitely glucosinolates have very good health value. In high amounts, there is a problem.
Okay, sir. I hope Charu ma'am got the answer. And there is one more question from Sakshi Gambar. She's asking, what was the reproducibility and rep- what was the reproducibility of data with various trials and how you validate such data? Means on which parameters you decide that produced data is reliable.
So it is not clear which data she's talking about,
Which
whether she's
trial
talking
she is talking
about
about?
whether
Uh,
she's talking about the biochemical data or about the genotype data. So the genotype data, um,
hmm,
because it is a very simple marker, it will not have any issue because very clear, just two bands, so there is no problem. Um and we don't rely on just one or two plants. So I told you we begin with some 200-300 individuals and then go on screening for different markers. And finally, we have some good number of plants, maybe 20, around 20, and those we take to the next generation. So from we have already selected 90% we have already is removed and 10% we take to the next generation. All of them means if there is a issue with some marker or some analysis, all of them will not be having that problem.
Okay, yeah, so
And finally, anyway, when we release a variety, definitely there is a much stricter analysis. So it is not just one sample will come and people will say that it is low glucosinate or low uric.
Uh, yes, Sakshi was asking about the reproducibility and genotypic data. You got the right thing. Dr. Sachin want to ask that how can plant genomics be strategically used to develop crop varieties with enhanced post-harvest traits?
Okay. uh, it is like whether we want so post harvest trait, again it is a one kind of trait. Now any trait if you want to improve, the process is almost same. It is like you begin with and if you are trying to use this conventional method, the genetics based method, then you have to have the donor lines. Now we are working on one crop, actually this chili, where we are targeting to develop varieties which are resistant to anthracnose, Coledotricum capsaicin. Now there, first important thing is you have to have a variation in that trait. So if you can find some, at least some lines which have resistance for that, then only one can go ahead. Once we find that variation is there, then second thing we try to find out uh, if you want to use this marker assisted approach, that where is that gene or what are the genes which control that? Are there few genes or are there many genes? Based on that, so first thing we want to get this answer. If there are few genes, and we definitely would like to find out their location, on which chromosome they are, and also develop markers which are closely linked with them, then we can use this marker assisted breeding. If there are many genes which are controlling this trait, for example, that happens especially in case of this yield related traits or quantitative traits, then marker assisted breeding is not the right approach. Then it will be better to use a genomics based approach, so what we call genomic selection, where we are actually taking data from across the genome and then developing a model. We call it a genomic selection model, and then based on that, we get the we kind of index. We form an index of individuals. So if there are 100 lines, we find out okay top 10 lines which have the highest desirable traits, and then we take forward. So it is like it is not a marker assisted approach, but it is a genomic assisted.
Okay, sir. Sir, one question from my side that there are different markers these days that we are using, right starting from the RAPD and SSR. That those days are gone. But what do you think? Which marker you now feel like find that it is the most efficient one that you will be suggesting for all crops?
So any crop, two high throughput markers are currently available. One is those which are based on chip, so microarrays based chips are there. Uh, there you have to be dependent. First thing, whether you are working on a very commonly or very important crop. For example, if you are working on rice, wheat, and maize, then you have the chips are available. But suppose you are working on a crop which is not that big a crop, or maybe the crop is not that important, chips may not be available. and then even developing a chip and utilizing it is a costly affair. Generally, 6 to 7 thousand per individual is the cost there.
Hmm.
Um, a much cost-effective method is this DD-RAD or genotyping by sequencing, where you can generate almost double of the data. Chip, whatever the chip can develop, you can develop at least 2 times of the data with almost one-fourth of cost. So less than 2,000 rupees per sample, you can easily generate 5-10,000 markers. No, that will depend again on the diversity of your samples. The material which is there, if they are completely homogeneous, then there is a problem. But then the problem will be with even chips also. But you can generate a very big data, very good quality data. People say the GBS has the problem of missing data, but when you have such a big data, you can easily throw out a lot of it. Still, you have a very good data and which is big, still big. So we are using since 2016, we are using genotyping by sequencing, and we ourselves. So it is like we don't send it to the company, so we generate ourselves, and we do all the wet lab plus bioinformatics part. um, in fact, you may not be aware, we are the only university who also provide services in this. We are the only university in India who provides services on genotyping by sequencing.
Okay, sir. So, one question from Dr. Chongtham Devi. She's asking, given the high heterozygosity, polyploidy, and long juvenile phase in fruit crops like mango and strawberry, how can advanced genomic approaches such as SNP genotyping and genomic selection enhance the speed and precision of marker assisted breeding in perennial fruit crops? Kindly share the insights.
Okay. Um, First thing I would like to say, and that is, you can say, a very negative aspect of it, is that in India at least, I don't find a long term program on any of the fruit crops which utilizes a, you can say, long term uh, vision. So mango, definitely mango is, you can say, the number one uh, fruit crop of India, because we have the greatest diversity in this crop, and we have very good varieties also. But if you see, those who are involved in this, uh, can you find a single research paper with genotyping by sequencing on mango from India? No, till date. Genotyping by sequencing was developed in 2011, and after 16 years, we have not a single paper from an Indian lab. Uh, all the research, if you get the published one, is either from USA or China. Recently, Dr. R.K. Varsne, he has published a paper, but again using the same data which was earlier used by those countries. So from India, we don't have any single data. So first thing, we have to, some those who have the germplasm, those who have the technology, those who have the, who want to develop or breed the crop, they all have to somehow come together, because the strengths are not at one place. It is very difficult to find all strength in one place. So always it will be better if we collaborate with different capabilities and develop a long-term vision. So especially the institutions who have got this mandate, they should have a long-term vision of developing and a genomic selection. So genomic selection cannot be done, especially in case of fruit crops, in 3 years or 5 year project. It has to be a very long term project. I just a few days back I was reading people who are working on citrus in US. They are working almost some 80 years. Means since 80 years they have the data. Do we have that? So that kind of long-term plan is required, and data um, repository has to be there. All because most of the time the data gets lost with the researcher. All that we do is just publish a paper, but the same data now with the genotyping by sequencing, you can always pool the data. Like if I have done genotyping by sequencing of 50 individuals, somebody has done another 50. So you can pull those two and you can get a bigger data. You can use the things to so-called machine learning and AI, and using that you can develop a good model, and your model will be as good your data and as the size of your data. So one has to try to increase the size of the data slowly. Like it cannot be done in a short time, but somebody has to begin this.
Okay,
So,
sir. So one last, okay, ma'am. There is one last question from Dr. Charu Gupta, ma'am. Sir, can we use plant genomics to increase or improve the phytochemicals concentration also?
You can. You're talking about this perennials or annuals. You can. Perennials breeding generally it involves only selection. Like you don't have this option of having so many rounds of crosses, because one round of cross then wait for another 5 years, because after 5 years only that seed will means give another seed. Um, at least in case of mango and this citrus, sometimes it will be much more. Not 5 years or maybe 8 years. So different. So that is why I'm saying those who are interested in this, they should come up with a long-term vision and means a collaborative approach.
Okay,
thank
sir.
you, Dr. Shashi, for such an informative talk. Sorry, I missed part of your talk. Did you talk about the Bhoot Jalokia also?
No, no, I didn't talk about Bhuj Jolokia. Bhuj Jolokia, uh, we have done this first thing, the genetic diversity analysis of Bhuj Jolokia. Bhuj Jolokia, people think it is one variety, but it
No,
is
no,
not
no.
a
It's
it is
not a
not a
variety.
variety. It is a
Uh,
land race,
uh,
and we collected so many samples. In fact, 91 uh, individual samples we collected, and we found that
hmm,
it has pungency level almost similar to hottest chili, means hottest capsicum annum. So that is the lowest one, and from that some 10 times of that.
hmm.
So the Bhuj Jolokia does not have one particular, only one single quality. Different Bhuj Jolokia has
Okay.
different quality, and we are seeing that its pungency is getting lost with time, getting lost because of mixture, mainly the gene flow and mixture. Admixture is there, so we are losing that. Food Jolokia, also you can find some important genes such as anthracnose resistance in that. So we had earlier developed a uh, breeding population, a segregating population where we tested it, and we actually found that it has good resistance against the um, anthracnose.
Okay. So, do you really think it is in the should we really like depend on the biodiversity and for the high nutraceutical crop, or should we go for the breeding, the genetic breeding or gene editing or something like that?
See, the available genetic diversity,
Hmm.
though means nature is making the diversity, but that is not for means targeted to our use.
Hmm.
It is like the you can say the survival of the fittest that mainly governs that development. But
Hmm.
our objective can be that a variety has means should have a particular set of characters that we cannot get ready-made within the nature. Definitely, we have to do some intervention so that we try to bring one after other the traits, so that so we have to do some integration kind of thing.
So within the species, instead of
within this,
doing molecular breeding, you go for the gene editing within the species.
see, gene editing again.
No, so for example,
it is same
like
thing.
yesterday Dr. N.K. Singh was presenting and he said it is exempted from the regulation actually. Gene editing within the species.
Yes, because no, gene editing even means gene editing happens always within species. It is like just changing one or two bases.
One or two
Now,
bases. Yeah.
that is why it is out of this purview of this regulation.
Regulations.
Because one cannot tell whether it is really edited or not. You can develop the same thing using mutations. So that way, but the thing is that if you are targeting it for a trait which is a simple trait, then gene editing is okay. But if you are targeting for a complex trait, then genome editing may not be the answer.
Just for example, glucosinolate.
So glucosinolate, you can still develop it through genome editing.
But there's so many multiple genes. I don't know how many genes.
No, but multiple means 2-3 genes. So we targeted only 2 genes. So that way it is possible. But suppose you are talking about yield or talking about concentration, I mean some height or concentration of something which is a quantitative trait, then it may not be possible.
Okay, so you mean to say that it's not good for yield improvement? Do you mean to say that?
That is a different issue. I mean, see, by gene editing is a kind of mutation. Gene editing is very same as mutation. Only difference is here you are mutating in a very precise way. If you are treating with say compounds called EMS, ethyl methane sulfonate, it can make changes anywhere. And then you have to look for the changes which you want. Here you can target the changes in a very precise way, and then it is up to you how do you want to use that mutant. After that anyway you have to do this background, this thing. So you have to develop the background. I give an example. Let me give an example. So suppose we are talking about a horticultural crop like mango, and if we want to change the vitamin C content or maybe vitamin A content in this, so there must be a gene. First one has to find out. First one has to find out which gene, if changed in a certain way, can lead to increase in vitamin A. So that is actually a kind of something which is lot that will require a lot of this. Let me just, my phone battery seems to be getting exhausted. So with the first thing you have to do is you have to find out which particular gene, if changes in a certain way, will lead to that particular change which you want. The second is, once you have changed that gene, you cannot change both the copies. If it is a diploid individual, you cannot change both the copies at a time. You can change only one copy at a time, which means you will not get the effect because the functional part is still there. One copy of function is already there. One copy of function you have changed by genome editing. Second copy is there. So that is not going to give you the effect. You have to make a selfing or crossing. Now, problem with this fruit species like mango is that once you make a cross, all genetics gets changed. It is a highly heterozygous class. Somebody told the heterozygous individuals this is the problem. So if you make a cross in heterozygous things like mango, you cannot get that variety back. तो अगर आप आम्रपाली का सीड लेके लगा दें तो आम्रपाली नहीं मिलेगा आपको, because during the seed formation lot of reshuffling of genes has happened. So that is the big problem. with genome editing if you want to use in mango. In case of crops, it is easy because you can self it and you can get the functional.
So annual crop is easier.
Yes.
Good. Thank you. Thank you so much. Very interesting.
Nice. So thank you so much for giving me this opportunity.
Thank you for accepting our invite. Thank you.
Thank you for sharing your valuable insights and experience with us. Thank you, sir.
Thank you. So we'll try to collaborate if possible, if there are some opportunities.
Yes, I will. As we discussed, we will definitely.
Yes.
Thank you.
Okay.
Okay. Thank you, sir. Before proceeding forward, I want to inform all participants that a feedback form link and an assessment form link will be shared with all of you in the chat box at the end of the session. So please attend whole session and participate in the assessment and feedback. Okay, now I'll be introducing our next distinguished speaker. It's my great honor to introduce our next distinguished speaker, Dr. Jagesh Kumar Tiwari, sir, Senior Scientist, ICR, Indian Institute of Vegetable Research, Varanasi. Dr. Jagesh Kumar Tiwari, sir, before that he has worked at ICR, Central Potato Research Institute, Shimla, during 2008 to 2022 on potato biotechnology, including pre-breeding, tissue culture, somatic hybridization, allele mining, mapping, molecular markers, functional genomics, and varietal development. He had a postdoctorate from University of Adelaide, Australia, under the Postdoctoral Endeavour Research Fellowship, Australia. He has significant contribution in development of interspecific potato somatic hybrids via protoplast fusion using non-crossable wild species Solanum cardiphyllum and Solanum panisectum and Solanum tuberosum to widen the genetic base of the cultivated potato for late blight resistance and potato virus Y resistance. He has established genetic stability in vitro plants, identified SSR for germplasm varietal identification, marker assisted selection, identified novel genes and microRNAs for nitrogen stress in potato tuberization and late blight resistance using transcriptome sequencing, and sequenced whole genome of dihaploid potato, that is C13, and glasshouse potato aphid, that is a common name is foxglove aphid. He has contributed in the development of 4 potato varieties, Kufri Neelkant, Kufri Jamunia, Kufri Lohit, and Kufri Uday, and 20 potato genetic stocks with ICR NVPGR New Delhi for unique traits, and he has been granted with 1 patent also on aeroponics nutrient solution and applications thereof. He has published many research and review articles and 6 books also. So the list is very long. I would like to inform that he also has a lot of awards and honors like ICR Lal Bahadur Shastri Outstanding Young Scientist Award, ICR Hariom Ashram Trust Award, Postdoctorate Endeavour Research Fellowship Australia, and CPRI Dr. Ramanujan Award also. And fellow also, he is a fellow of National Academy of Agricultural Sciences, Indian Academy of Horticultural Sciences, Indian Society of Vegetable Science, Confederation of Horticulture Association of India, and many more. Sir, we welcome you. The title of the talk of Sir is Emerging Trends and Innovations in Varietal Development and Post-Harvest Technology and Management in Vegetables Towards Sustainable Food Systems. Sir, we welcome you, and it's over to you.
Thank you very much, ma'am. Am I audible?
Yes, sir, you
Thank
are
you,
audible.
sir. First of all, good morning and welcome to MIT University, sir. Thank you.
Good morning, good afternoon to all of you.
Good afternoon. Good afternoon, sir.
Is it audible now? No issue.
Yes, sir. Yes, sir. You are audible.
Okay. Thank you very much, ma'am, for introduction about me. I'm just a learning scientist only. And so before this IIVR, I was at CPRI Shimla for 14 years, as you said. So currently at Indian Institute of Vegetable Research for the last 4 years, and mainly working on tomato breeding and genomics. So moving from potato to tomato. So may I share a slide, ma'am? Is it visible, ma'am, please? Is it visible to you?
Yes, sir, it is visible. Actually, Dr. Namika, the sound is not coming.
I see is muted. Okay. Okay. Thank you so much. So I have been given 40 minutes time, I think, 40 to 50 minutes. So I will be trying to summarize our work at IIVR Shimla, IIVR Varanasi. In fact, we are focusing more on development of varieties and then post-harvest technologies. So emerging trends and innovations in post-harvest technologies and management towards sustainable food security. So I will be talking in your this 5 days faculty development program. Talk on vegetable this varietal development and post-harvest management in our vegetable crops. So first of all, this vegetable, we know every day we are using total horticulture production, as you might be knowing, it's surpassing like anything. Currently, it's more than 350 million ton. So which is a huge record. Every year, it's going day by day. Out of which our vegetable is more than 50% of production it's covering. So vegetable is a key source of our phytonutrients, as we know. So target for our future Viksit Bharat 2047, we can say area is not going to much high. That's up to 17 hectare, but production we have to reach up to 457. So this production of vegetable crops, especially I'm talking, currently it is nearly 212 million ton and has to reach more than 450 million ton. Per capita availability of vegetable, we know it's nearly 300 gram per day per capita, but it has to increase to 500, more than 500 grams per day, so that we can meet our population requirement. And population is also going to be increasing day by day. So our target is to meet this 1.7 billion of population by 2047. So as we know, this NIN recommendation of 400 to 500 gram vegetables per capita per day, it's our requirement. So as you can see, this graph of vegetable states where Uttar Pradesh, West Bengal, Madhya Pradesh, these are the leading states in India having a very high production rate. So this is a nutshell about our vegetable crops out of total horticulture of 350 million tons. So looking at this vegetable as a journey, it was at Varanasi. This All India Coordinated Research Project was started in 1971. Then project directorate was there at Delhi. Then it was shifted in 91 at Varanasi. And then our institute, this Indian Institute of Vegetable Research, came into full fledged as the Indian Institute of Vegetable Research in 1999. So after 1999 or 91, we can say, as a PDVR, 35 years of journey of this institute has. And founder director of this institute was Professor Kallu Gautam Sahab. So you might be knowing those who are vegetable scientist or any crop scientist. So Professor Kallu Gautam was a founder director of this Indian Institute of Vegetable Research. So our target is to meet this requirement of vegetables. So about post-harvest losses in horticulture crops, as a vegetable, we can see this as per the annual report of Ministry of Food Processing India. Vegetable is nearly 4.58 to 12% of losses as per the CFET study. So nearly up to 15% it has been reported. And globally, this all food loss is about 15% in the world. And but in developing countries, it's going to be nearly 45% total fruits and vegetable losses. So our challenge is very huge. So our processing sector has challenge of supply chain, infrastructure, then inadequate linkages between production and processing, seasonality of operations and low capacity utilizations, supply chain management, inadequate focus, so lack of production development innovations. So these are the major challenges what we are facing as a vegetable and a very perishable commodity, in which tomato is also having the very huge losses in terms of post harvest nearly 12% or 13%, and so this another study data of similar type of the CFET record. So tomato is nearly 13%, others so up to 14-15% of post harvest losses. So can be this if you compare with our developed nations like Brazil, USA, where processing is being done at huge scale. Like more than 50, 70, 80% of processing is being done in the developed nations, but we are very less, nearly 2% of processing in India. And globally, FAO says 30 to 40% of post-harvest losses, as I said earlier. So there are a couple of factors responsible for this maturity, standardization, harvesting systems, pre-cooling. So these are the major factors. And before that, I also said the challenges in this post-harvest management in vegetable crops. Side by side, if you look at the processing countries, where especially if we talk of tomato as a major processed product worldwide, having the more than 45 million tons of potato is processed as per the world processing information, and where China, California, USA, Italy, Spain, they are leading one. And unfortunately, we are at very low in processing sector. So globally, if you look at this figure, this from WPTC, so where China is and where India is 0.16 and China is 10. So we in million tons of processing industry. So until unless we focus on processing, so we have to. So these are the different groups of world tomato processing worldwide, North American, Mediterranean regions, these other countries wise groups where tomato is being processed, and we are using in different forms. So why the processing is low in our countries? So based on our specific requirement, I have summarized this one: supply issues and variety issues, especially for lack of processing varieties, which is very much important. Short supply window, especially North Indian conditions, supply window of this tomato growing seasons is limited due to high temperature, as well as coupled with high humidity in rainy season crop. Quality and technologies, economics and imports, infrastructures. So this is one of the major processing sectors which has to be fill the gaps using the cold storage technologies. So that's why impact on farmers and prices, it affects farmers often face price fluctuations, especially in top crop, you can say tomato, onion, and potato. So how to stabilize the prices in combination with technology integration, as well as this post-harvest management technology or value addition. So these are the different forms which are already working in our India, Hindustan Unilevers, different forms are there. These are the common products which we are using in our daily routine. And major states which are these companies are established, these Andhra Pradesh, Delhi, Maharashtra, Punjab, most of them are southern part of India, and a few are in north part of India, where tomato supply can be regulated and can be ensured to run this business. So this is benefits of different vegetable crops, as we say, purple vegetable crops like beetroot, red cabbage, eggplant, that is brinjals, having the high antioxidant properties, red, that is capsicum, radish, tomato, having different antioxidant compounds like this lycopene in tomato, this capsaicin in capsicum, this orange. So these are the different colors having the different advantage at our nutritional or phytonutrients level for the health purpose. Different vitamins and minerals having rich in the vegetable crops, this sodium, iodine, vitamins, phosphorus. So I mean to say this summarization of vegetable having rich source of nutrients for our daily routine. So this is the journey of my institute. As I said, it started from AICRP vegetable crops, that is All India Coordinated Research Project on Vegetable Crops in 1971. Then it was converted to PDVR, then PDVR at Varanasi, from Delhi to Varanasi, and then now full-fledged institute for the last 35 years. So I'll be coming to these achievements of in vegetable crops. This is the National Institute working on all vegetable crops except potato and onion. Potato is major done at Central Potato Research Institute at Shimla, where I was earlier, and then onion is done at Rajguru Nagar, where near Pune. Directorate of Onion Research (DOGR) is there. Besides onion and potato, this Indian Institute of Vegetable Research undertake research activities and development of varieties and technologies for all vegetable crops. Nearly 42 vegetable crops we are covering. So these are the multiple challenges aligned with our sustainable development goal, that is to meet this no poverty, zero hunger, good health and well-being, quality education, gender equality, and in all climate resilience. So that's why in all crops we can see multiple disease resistance. This is the top most priority in all these vegetable crops, major vegetable crops I have listed here. So multiple disease resistance, I mean to say in tomato, if we say an example, that is TLCV, early blight, late blight, high temperature tolerance. This is the most crucial nowadays in the era of climate change. We are also working under the NICRA, if anyone knows, National Innovation on Climate Resilient Agriculture. This is the global, I can say in all ICR institutes, this NICRA project is going on, and very high, this climate resilient work is undertaken in, especially in tomato at IIVR. Similarly, in brinjal, multiple tolerance, high temperature tolerance, chili also multiple tolerance to thrips, black thrips, white thrips, anthracnose, viruses, high temperature tolerance. Quality is nowadays topmost priority also there. Okra, this is one of the key crop having the YVMB, OE, CLCV, jazz seeds, cowpea. So these are the different crops, carrot, cucumber, bitter gourd. Underutilized crops, this is nowadays we call it as a opportunity crop, which are a very good source of nutrients, vitamins, phytominerals. So that's why utilization of underutilized crops like water spinach, wherein one variety is there, I will show later part of this study. So these are the crops where we are targeting our, if briefly we can go for the priority areas like biotic stress, abiotic stress, then what are our themes we are targeting for the climate resilient agriculture, where development of varieties and hybrids, these are the major our targets. Rootstock breeding we are undertaking. Rootstock are very important nowadays to mitigate abiotic stress tolerance. I mean to say waterlogging tolerance, like in rainy season, heavy rainfall, incidental rainfall happens this year also in September, in August and October. So that's why rootstock breeding is important, and we are working on that, especially on tomato and brinjal. Tailor-made varieties, genome editing, this is one of the flagship project we are working here for the different traits like multiple disease resistance, shelf life and processing quality in tomato, chili, cucumber and melons. 7 crops we are working on genome editing. Quality seed production, this is one of the important parameter for our farmers to supply the quality seeds in all vegetable crops, where as per our mandate and based on the indent received from the Government of India. Safe vegetable production, that is use of biopesticides, we can say. And that's why pesticide residue analysis being done, and we have the dedicated laboratory for residue analysis with LCMS. Then eco-friendly management of pest management. So these are the we are working on management and breeding aspects. Input use efficiency, mainly for the nitrogen use efficiency, we can say in tomato we are working precision farming with the use of drones and decision support systems. Organic vegetables, this is the parameter. This is as a Government of India, this is our natural farming and organic farming is our key. Year-round cultivation is one of the very important aspect nowadays because availability of tomato is very crucial, especially in North India, where we can't meet year-round cultivation of tomato. That's why we have to use either hydroponic, aquaponics, and potato structures. So these are some of examples of our climate resilient varieties, we can say. The Kasi Tapas, this is one of the hybrid of tomato that is resistant to high temperature, and Kasi Adbhut, this is also high temperature resistance hybrid of tomato. These were released last year by IIVR. These are the product outcome of NICRA project, as I said earlier. Kasi Rituraj, this is a climate resilient hybrid of radish. Kasi, this Utsav of brinjal. Kasi Baramasi, this is a very good variety of palak, that is spinach wheat. Kasi Manu, I was telling as a year round, this is water spinach. Ipomia, this is a genus of Ipomia aquatica. So these are few examples of our varieties which are climate resilient at national level, and some of our varieties were dedicated to the nation by Honorable Prime Minister. Kasi Baunisem 207, Indian bean, and Kasi Subra as a bottle gourd. This we call it as a gutka type, this bottle gourd. This is mainly suitable for transport purpose. Kasi Lalima in okra, this is phytonutrient rich, especially anthocyanin rich some vegetable varieties like Kasi Baunisem. Baunisem means dwarf sem, Indian bean, that is Dolichos bean. Kasi Bangli, French bean. This is Kasi Krishna in carrot, Kasi and Puna in winged bean. So these are few examples of nutrient rich vegetable crops. Processing and others. This Kasi Pratishtha in bitter gourd were released last year. And then spring, summer and rainy season, Kasi Parakram, Kasi Sahisnu, these are the okra variety. Besides, many of genetic stocks at our institute have been registered with NBPGR New Delhi. Suppose for this tomato, 5 lines are there, out of which one is H8878S1, that is resistant to early blight, root knot nematode, tomato leaf curl virus, and high temperature tolerance. This is one line having the multiple tolerance and being used widely in breeding purpose. So this is just a image of overview of this H8878S1. I think you might be knowing this grafting technology in fruit crops. This is called Brimato. Nowadays, if you Google Brimato, this is brinjal and tomato. So in single plant, you will get brinjal as well as tomato fruits. So this is nothing but grafting technology, where brinjal and tomato both are grafted on the same rootstock. So this is a certified technology by this Indian Institute of Vegetable Research and being widely used by the farmers, especially for cultivation of this Brimato. actually this rootstock breeding is a part of rootstock breeding, and we are using rootstock as a abiotic stress tolerance genotype. In other biomolecules, we have developed biomolecules and then LCMS facility, as I said, for residual analysis of vegetable crops. So biomolecules are being used to mitigate the chemical fertilizers, and so that we can enhance soil strength as well as biopesticides. So CASI, CRB7, and all this BC6, AJOBC, these are the bioformulations at our institute and being licensed to different firms. So I was talking about these processing sectors. These were old varieties developed earlier, 20 years back or before that. But nowadays, as per the current requirement of processing industries, these old varieties and they do not fit nowadays, especially in terms of pulp recovery or TSS and other parameters like biochemical parameters. So these are the few imported lines which we have being used in breeding purpose currently. These were imported from World Vegetable Center, Taiwan, and special having the processing attributes. Besides processing, it has also tolerance to various biotic stresses like tomato leaf curl virus, bacterial wilt, and so on. So out of which these are the few genotypes which we are using in our breeding to develop processing varieties.
Good afternoon, sir. Is it possible if you can finish your presentation by 2:15, because Dr. Shubhra has to leave by 2:45. So she has requested if
Okay.
she can start the presentation by 2:15. I'm sorry, you can continue after at 2:45 the remaining slides if possible.
Okay, so within 10 minutes or 5 minutes?
Within 15 minutes, sir. We have
Okay.
15 minutes.
Okay, right.
Thank you. Sorry, sir.
Okay, no issue.
I'm sorry because
No.
there's some problem. She
Okay,
has
no
to
worry.
leave for some unforeseen circumstances has arrived.
No issue, ma'am.
Okay, so sorry, sir.
So these are the few new varieties. If anyone is interested, they can note it. These are the hybrids. Pusa Prasanskrit. It was developed by IARI New Delhi, Indian Institute of Agriculture Research New Delhi. This is Pusa Prasanskrit, the oval shaped tomato hybrid, having tolerance to disease resistance. So this is having the very good product recovery. These are the two hybrids developed by IHR Bangalore, Arka Apeksha and Arka Vises. These are very good hybrids developed by Dr. A.T. Sadasiva sir, having the very good recovery of pulp as well as the TSS and being commercialized to the private firms. At IIVR, we have developed a Kasi Shrest, this for the processing or dual purpose type hybrids, having the good pulp recovery as well as the yield. More than 1,000 quintals of yield is also observed in these hybrids. Value addition, this is our at Indian Institute of Vegetable Research, we are using this moringa powder or modified atmospheric storage facility using different chips, this bitter gourd chips, moringa powder or different cookies. Different products are being done at to add the value in vegetable crops. So these supply chains, it's harvesting, trimming, grading, packing, storage, transport, wholesale. So this nothing what we know everybody, but we have to minimize the post-harvest losses at different places. Operation Green was launched by the government of India in 1920, and these were the tomato, onion, and potato. These three crops were covered under this to minimize the post-harvest losses by Ministry of Food and Processing. So these are some typical parameters for this processing industry, like high TSS. This is very crucial. Color value, that is more than 1.95, you can say. Acidity. So these are the different parameters. Along with that, high yield and resistance, this is very much important. Jointless is a key parameter for processing. These are some breeding help we can say for the breeders, these are some genes which must be in the processing varieties or genes. So these are some wild species which having the tolerance or resistance or quality traits which have been to be utilized in our breeding purpose. These are the few breeding parameters like fruit shape, oval, lycopene content, fruit firmness, TSS, pH, lycopene content, dry matter, this pulp percentage, pulp recovery, shelf life. So these are nowadays our ministry is also focusing on the increase the shelf life of tomato, so that how we can do it. So to 20 days, 25 days, more than 15 days at the normal room temperature we can keep. So the technology we are working on it to enhance the shelf life of tomato. In briefly, I will go. I said some genomic resources is being used. We reviewed this work to enhance genomic tools in tomato, and just genotype by sequencing. We have published one paper just recently in tomato using more than 72 lines of tomato by the GBS. These are pan-genomes of tomato genome, which is already available. And tomato genome sequence is nearly 900 MB, and more than 1,000 of tomato genome is available in database of NCBI and being used by the tomato breeders. So GWAS study, as I said, being used to mine the genes and for development of markers linked to the key traits. So genomic selection we are using to identify for the complex traits like processing to identify some markers, SNPs, so that we can breed for high TSS or high lycopene content. Genome editing, as I said, we are also working at IAVR for in tomato, that is for enhancing TSS and tomato leaf curl virus resistance. These are the summary of genes being used in tomato for different traits like plant growth habit, biotic stress, fruit maturity, abiotic stress tolerance. These were published earlier. So these are the two genes we are working in case of tomato, TSS and lycopene content. So these are the successful examples. Recently in the Australia also purple tomato have been just released now, and before that in Japan also GABA rich tomato was released. So initiatives, current initiatives, cutting edge technologies which we have to work nowadays: genome editing, genomics assisted breeding, then climate smart horticulture, which includes different grafting, precision farming, environmental horticulture, digital horticulture, IoT, Internet of Things, integrated management, pesticide detection kits. So these are the new initiatives being undertaken to enhance the vegetable production, productivity, as well as the health point of view. Nano fertilizers, integrated aquaponics, high-tech nursery, crop diversification. So these are the things, these are the success stories of vegetable crops which are reaching to the farmers. We can say in case of pointed gourd, in case of okra, in case of palak and different mushroom cultivations. So these are the success stories. So if you look at our institute roadmap for the 2024 to 29 and then to 47, so this is germplasm conservation. Currently we are holding more than 6,800 of vegetable accessions, pre-breeding and genomic gene stacking, climate resilience, input use efficiency, crop production technologies, then seed availability, quality seeds, post-harvest management, diagnostic tools, and genomics selection, genomic tools for the vegetable improvement. So this is similar to that strategic approaches we are using. IoT, precision management, climate resilience, supply chain coordination, market intelligence, diversification and innovation, sustainable practices. Ultimately, policy framework is one of the key for improvement. So these are the challenges in our vegetable crops, strengthening of research on tomato processing, we can say, for example, through public-private partnership, then year-round cultivation of to avoid the seasonal gluts, and minimum support prices and government policies to intervene these. So this is in nutshell about my presentation on the vegetable improvement. I can say. So this is all about from my side, ma'am. Thank you very much for giving me opportunity to talk on these vegetable crops and work at Indian Institute of Vegetable Research, Varanasi. Thank you very much. If any query, I can address.
Thank you, sir. This session is open for question and answer. If anybody have any question, you can put it in the chat box, or if you want to ask directly, you can raise your hand and ask.
Good afternoon, sir.
Good afternoon.
Sir, I have one queries like just now, sir has shown pictures of grafting with tomato and brinjal. So since tomato and brinjal, they belong to the same family, but I just have one question, that is, can this tomato and brinjal grafting can be sustained for the long-term production?
Yes, at our institutes, this is one of the key technology nowadays to mitigate the abiotic stress tolerance, especially waterlogging condition. As in North India, we cannot plant our tomato in open field during rainy season. I mean to say in July. So when rainfall is there, so for that, for abiotic stress tolerance or for soil-borne diseases, we are using brinjal rootstock having tolerance or resistance to those pathogens or those stresses, and we are grafting tomato, and that is very fantastically. We are getting production also, but seed is not available for that. Only plants are being sold. Okay, grafted plants like other fruit crops. So you have to raise your grafted plants every time for that. So it is nowadays already proven technology, brinjal rootstock and tomato sign. So this is
Sir, one thing also, it will not depend upon the varieties compatibility.
yes, it will depend definitely, because all genotypes, all varieties are not. Suppose we have one rootstock that is EC10056 of brinjal. It has tolerance to all biotic abiotic stress. But all sign of tomato, all cannot be compatible having the good yield. So you have to decide, we have to standardize those rootstock and sign compatibility. Then only we can.
One last questions from my end. Like the sir has mentioned about the wing bean, and even I saw with Rakesh sir, Roli sir also, he used to put up in Facebook like doing breeding. Rakesh Dubey sir, I think sir might be knowing. So in Manipur, this wing bean we generally consume as a salad purpose, especially during this winter season only. Sir, when we are consuming as a salad purpose, we always think about the tenderness. So after 1 week or hardly after 7 or 8 days, like from the flowers, it will start consisting of the fiber and all. So according to the Manipur situation, what are the climatic conditions and what genes or something we need to incorporate or what, so that we can get it tender as well as throughout the season, the harvesting period also we can be extended.
It's very good. I think this Northeast is very rich source of genetic biodiversity. And
Yes,
if
sir.
you know Dr. Dubey already, he's working on that aspect. So
Yes,
you can
sir.
be directly in contact with him, and then we can evaluate those lines there also.
Okay.
And to get because gene to interaction is there, might be already known. So genotype environment interaction is there. All Bhut Jolokia, this chili genotype,
Yes, sir.
not
King
as pungent in our condition, but it is pungent in your condition.
Yes, sir.
So, like that, this winged genotype, what do you like to grow there? You can contact with him or to write.
Yes, sir. He is my professor. He taught me BSc.
That those lines you can evaluate there also, because here more than 200 winged bean lines they are maintaining. So that can be collaborative work, and that those can be evaluated at your place.
Okay.
So
Thank you so much, sir. Thank you so much.
Thank you.
So one question from my side. For example, if we talk about like we are talking about tomato, brinjal, and all, we know we have a lot of varieties, hybrid varieties, and we have done a lot of work on these crops already. If we talk about orphan crops, and we are talking about molecular breeding these days, we know that we have not identified all of the important genes like QTLs or all these things in those particular orphan crops, and we know that all those orphan crops, they are much more higher in nutrition as compared to the crops that we are consuming.
Right.
So why is it that our researchers are not much, working on these, like on the genome sequencing or all these things to, particularly for the orphan crop or, less utilized crops?
Ma'am, it's very good question. Nowadays, it's called opportunity crop. Our honorable DG says now it's opportunity crop. Now they are not orphan; they are opportunity for the future, having the rich source of these nutrients and all abiotic stresses. So we must work on that. As our in slides I showed opportunity crop or underutilized crops. They includes I think more than 10 crops are there. And at nodal or National Institute of our government of India, we are working on that. In winged wind, ma'am just said our Kasi and Puna is one variety have been released by State Variety Release Committee. Kasi and Puna in winged wind only, and other crops also. Kasi Manu I said in water spinach. Water spinach that is very climate resilient genotype. So we are working. But yes, that is also right.
The water spinach is a pohi, sir? It's pohi or?
Not poi, that karamua we say earlier. It's
It's karam sag.
तालाब में होता था, but
A
now
kalam sag, no? That
Karam
I think
sag,
we
yes.
developed a proposal on that one, though.
Yes,
It's the
ma'am.
same
Karam
thing,
sag.
no?
Uh-huh.
So we developed one proposal. Maybe we will discuss with you, and then we will like to take it further with you for that project which we developed earlier. Kalam sag par. So,
Karam, that is very.
That was Malabar spinach, sir.
Malabar spinach.
Malabar is a different.
No, no, it is water
It's
spinach.
different.
Water spinach.
Water spinach one.
Okay.
No, sir. Yeah.
Okay.
Okay, sir. So very good presentation, sir. And I think we will be in touch with you regarding the possibility of the collaborations, internship opportunities, student exchange, or something like that. We have it's an ICR institute, no? IIVR.
Right.
So we have an MOU with ICR, and so there should not be any problem in collaboration. So thank you so much, and we'll look forward to further collaboration, sir. And Dr. Ramika, all the questions are taken up?
Yes, ma'am. Yes, ma'am.
Okay, thank you, Doctor. Thank you so much, sir.
Thank you. Thank you.
And I'm sorry for cutting you your time, sir. I'm really sorry for that.
No, please don't say sorry. It's fine. I completed.
Yes, we will invite you for at some other point to have a detailed presentation from you.
Thank you so much.
Thank you, sir. Whenever you are in Delhi, please let us know.
Okay, thank you.
Yes, sir. Thank you. Thank you.
Okay, sir. Thank you for sharing your valuable insights and experience with us. Next, it's my honor and pleasure to welcome and introduce our next distinguished speaker, Dr. Shubha Chakrabarti, ma'am, Professor, former Director, NIPGR, New Delhi. Dr. Shubha ma'am she has obtained her PhD in plant molecular biology from Jawaharlal Nehru University after a brief career as research scientist at the Center for Plant Molecular Biology, Jawaharlal Nehru University. She joined National Institute for Plant Genome Research as one of the founding faculty and has been continuously ascending the ranks ever since. She worked as visiting scientist at the Yale University, USA, trained as molecular biologist and biotechnologist. She got interested in protein science and was instrumental in initiating and establishing plant proteomics and translational genomics research in India. The major research interest of her laboratory is focused on nutritional genomics, translational genomics, plant immunity, multi-host resistance proteomics, and plant biotechnology. The targeted research in the field has direct implications in plant and human health and vital to food and nutritional security.
हमारा ये है हमारा ससुराल में सबका जल्दी ये होता है मायके में नहीं कम है लेकिन मेरा अभी तक हुआ नहीं कभी एक आधा दिखते हैं
अर्पिता मैम
नहीं दिखता नहीं है
अर्पिता मैम प्लीज म्यूट योरसेल्फ. Okay, I'm sorry for the disturbance, ma'am. The awards and fellowships she has is Fellow of Indian National Science Academy, a recipient of Vivek Jyoti Samman Lifetime Achievement Award, Young Scientist Award, and there are so many awards. Dr. Chakrabarti ma'am has played a key role in advancing plant biotechnology and improving nutritional security in India. Ma'am, we welcome you. And please share your valuable experience, knowledge, and guide us new budding researchers and teachers, please. Ma'am, it's over to you.
First of all, at the outset, I would like to. Am I audible?
Yes, ma'am, you're audible.
So I would like to thank the organizers and particularly Dr. Nutan Kaushik, who is a very good friend for many years now, I think, and all of
Thank
you.
you. Thank
And
you. I must thank you because you
no,
have accepted despite your difficult time.
no.
Thank you so much.
And
Thank you.
I want to apologize to my previous speaker to cut short his time because actually I had a sudden demise at home, and therefore, because I committed to Nutan, so I thought I'll come and do justice to this. So I just came back from there and I have to rush there. That is
Thank you
why.
so much for
Uh,
honoring. Thank you so much. I'm really
yeah.
sorry for that loss.
No, then that's fine. So, only thing is that before I start, I would also like to tell to the audience that although this is on post-harvest technology and the vegetable crop, but because of this, all incidents was going on. So, I have kept my talk on rice. However, it will be with a post-harvest quality trait, and that trait, whatever discovery we have caught in rice can also be translated to the vegetable crop because I'll be speaking about two traits. One is the aroma, which is very important, and that aroma is also there in many of the vegetable and fruit crops. And other point that I would be talking is the protein, which is a very important aspect of any kind of nutrition, whether we are eating vegetable or a cereal crop or a legume crop. So somehow I'm going to do justice on that, Nutan, and I thought to give a talk on
Perfect,
tomato.
perfect, perfect, Dr. Shubhra.
I could
It's
not
the approach which
get the time.
which matters, actually, not the crop.
Okay, so just allow me to share my slides. Just give me a
I thought
second.
you would be sharing some of your tomato experience.
Actually, I could not make my slide. Actually, I
No,
thought
no, it's
that.
okay. It's okay, sir, Dr.
Sorry
Shubhra.
about it. So, is my slides available? Visible?
Yes, ma'am, it's visible, but yeah,
Uh,
yes, ma'am.
I,
Please
so
do it on share.
bypass this private window picker. So I'm getting some message. Zoom.us is requesting to bypass the system private window picker and directly access your screen and audio. Allow or open system setting. Should I allow?
Allow is okay. Allow is okay.
Okay. So is it full screen now?
Yes, ma'am.
Okay. So I'm just uh, okay. Okay, so today I'm going to talk to you, and my title, as you are seeing, is Unfolding the Secrets of Seed Nutrient Dynamics Using Integrative Omics Towards Quality Trait and Nutritional Security. As Nutan was telling, that it is important that what kind of approach we are taking. So, in this whole work, we have been utilizing integrative omics where we are trying to map all the ohm together to find out the biological pathways or the cellular pathways which are related to the quality trait, which is so very important for the nutritional security. So I'm going to touch upon that. Before that, many of you might be knowing, and those who have never come to this beautiful campus, please do visit us. I'm working right now as a distinguished scientist, just relinquished as the former director. So this is National Institute of Plant Genome Research, which is now a BRIC institution. So, to set the tone, I would say that my lab works on two major areas. One is plant pathogen interaction because, as you all know, that pathogen stress accounts for more than 70% crop loss worldwide. Not only that, we have seen during COVID time, infectious disease accounted for 36 million deaths in 2021, and top five included COVID and many of the tuberculosis and many other diseases. So, one very important thing is that is One Health that we talk today, and we have been working on one such pathogen, which is Fusarium, which can infect not only plant, as you could see over here, it infects the plant through the root, even to the tomato and many other plant. At the same time, it can also infect human and animal. So in that case, it is a multi-host pathogen problem. We just got a paper last month out in JPR on that where we have talked about it. So anybody interested can Google and get that paper to know more about multi-host resistance. The second aspect that we have been working from my almost PhD days is something around on malnutrition or overnutrition, where the nutrients and the post harvest quality is very, very important, as 13% of world population suffer from food and nutrition security. We have 35% of all child death which comes due to the malnutrition, 11% of global disease burden. And we also are seeing that the kind of food habit that demographic change is bringing forward in our diet pattern, even in India, is that lot of, the food which are junk food that we call, and that is where we are getting 40% of Oceania population is getting obese, and India is also joining hand, and where the kind of NCDs which are non-communicable diseases like diabetes, like many other diseases which are very, very important. So with these two aspects in mind, we have been working towards these two areas. Now, with this, our focus, as I said, plant health and multi-host resistance, protein nutrition, anti-nutrient management, post-harvest quality, and we use system science to understand the basic biology, pathways, and then once we understand that, we try to clone genes or try to get protein and take them for application purpose, so as to make food and nutrient safe crop plants. And the plants that we have been working are rice, chickpea. We are also working on tomato and potato, which are the vegetable crops. Today's talk. Then we are also working on jute, lathyrus, spinach. Spinach is another one that will be a vegetable, and also some other legume crops. So with this, I'm going to talk to you today about the trait which we have found out in the rice, but which can also be implicated in tomato, in potato, as well as in spinach, which we have been working in the lab. So I'm coming to first problem, which is protein energy malnutrition. As you could see in this slide, that protein deficiency syndrome, it's a syndrome. Syndrome means something where many of the factor makes one disease, and that is called a syndrome. So here it is a protein deficiency. This is a slide, comes from South Africa, where African population, where the children are very, very malnourished. I would like to remind you, the situation is not very different in India, even if we are talking about Vikshit Bharat and everything. If you see, look into this pie, 70% of Indian population annual child death comes from PM, which is protein energy malnutrition. That is the highest in Asia, which is quite I would say a shameful situation. And if you could see the current situation of India, India is somewhere which is like African nation. So now with that, if you see the UNICEF data and FAO data that tells, that WHO data, this is very recent 2023 data, it speculates that in 1990, although South Asia was always on top about the protein deficiency syndrome or protein malnutrition, but prediction, which is alarming when we are going to do meet 2047, but in 2044 you see where we will be standing in terms of protein malnutrition, which is very, very important aspect that we need to look into. And this increasing the good quality of protein in not only is important for cereal, but also is very important in potato. We already have done some work many years before in collaboration with Sarai Shimla, and on that we have made the potato which are having more protein, more amino acids. So
I think we lost the connection. You're unmuted, sir. Unmute yourself, Dr. Shubhra.
yeah, but I'm seeing Nutan your face. I can't see my slides anymore.
Can you share it again? I think it's unshared.
My God. Just a moment. Is it okay, Nutan?
Not yet.
In my computer, it is shared.
It is coming up. It's coming up. Yeah, now it's okay.
Okay, so if you see the protein content, although the rice, each part is very, very important, and as you could see, how much protein we have as a paddy, as a husk in the broken area, in the starch or in the bran. But if you see, bran is very high in protein content, but the one that we actually consume, that is a milled rice or the broken or flour, that is quite low, which is 7%, among lowest among the cereal. So that sets the tone that why we started working on rice now. Now, in that, if you see, we now are having a mission mode program which has been funded by DBT. I'm coordinating this program along with three of the ICR Institute, IRI, NBPGR, NRRI Cuttack, one nutrition institute of ICMR, that is NIN, and another is an hospital, that is SGRI in Bangalore. So the whole program is on national mission to improve nutritional quality of digestible protein content and quality in rice. And what system biology we are taking here is to decipher complex quantitative genetic architecture by identifying functional QTL, PQTL and MQTL from the genomic, proteomic and metabolomic study, which from where we get the key molecules which govern the protein quality and quantity in rice. And when we do that, we identify these as a basic science. We want to apply that to develop high yielding rice lines which will have digestible high protein and essential amino acid without yield penalty, which is very important for farmer perspective and for better nutrition with three-pronged approach. One will be MAP, that is molecular assisted breeding. One is genome editing, another is transgenesis. Now, the approach that I am talking to you, so we have been doing using the 90K SNP chip, which has been developed by Dr. Parida or at NIPGR and has been used in the whole rice mission program right now for last 4 years in the DBT funded program. Now we have also have a very high end proteomics facility in NIPGR in my lab, as well as a very high end quantitation using near infrared spectrophotometry to quantify protein, which can quantify more than 1,000 germplasm in a month time. And we have been using here a mass spec for the metabolomics as well. And then we integrate all this data, and this is the work plan that we have been following to come up with a high yielding and high nutritive value of rice. Now, when we talk about the nutrition is a trait which is governed by biochemistry. And therefore, what in this paper, which we have published recently in food chemistry, what we have done, because we will be expanding this into a 12,000 germplasm stored in NBPGR, before doing that, we wanted to set up all our genomics, proteomics, and metabolomics on one of the very, very popular rice variety, which is a very important donor variety for any breeding program, that is IR64. So what we did, we actually followed the when seed matures. As a botanist, you would be knowing that we go through a milky stage to a mature stage to a germinating stage. So what we did, we collected the samples from IR64. We then quantified the protein. As you could see, maximum protein we get in the mature seed. And then we also did all the amino acids, because the protein quality that I am talking to you is determined by the 10 essential amino acids, of which in rice, lysine is the most limiting, along with methionine, phenylalanine, and proline. So, therefore, we also measured the water content. At the same time, we measure the tannin and phenol, because tannin and phenol are some of the biochemical molecule which actually is inhibitory compound for protein nutrition. And with that, when we mapped the metabolomics, we found that starting from this methionine cysteine metabolism through many of the amino acid biosynthetic metabolism, then sphingolipid metabolism, primary metabolism, arachidonic acid, purine metabolism, and TCA cycle top the list for all this metabolic profiling data. So when we went deep into this data, along after doing this metabolomics, we also did a high throughput LCMSMS based proteomic study, from where we found out at these three different stages, 392 proteins were common between MS, mature stage, milky stage, and germinating stage. However, those stages also showed many different, like 28, 21, 54 proteins, which are, or 60 proteins, which are unique to each one of those stages. And therefore, they possibly, what we believe they possibly is doing specific expression. Now, when we also tried to map all them, we did a total network analysis by cytoscape, and in that case, as I'm not going to read out, but you could find out there are many translational proteins that came up in our data, which possibly is governing the whole translation. And when we get the protein, it is nothing but a product of translation which is coming from the mRNA. So with this, we then moved ahead and we tried to found out that where all these metabolites coming out from our metabolomics data are distributed. So what you could see is that some of the metabolites are, most of the metabolites are in cytoplasm, which is not very unlikely, because cytoplasm is the site within the cell which performs the primary metabolism function along with the mitochondria and chloroplast. And therefore, we see the first three topmost organelle where all these metabolites are accumulated are cytoplasm, mitochondria, and chloroplast. Some are followed by some other organelles that you are seeing. And this data shows you how these, let's say a cytoplasmic metabolite is connected with a peroxisomal metabolite, or a cytoplasmic metabolite is connected to somewhere into the nuclear metabolite, that you could see. So that means there is possibly a connection or kind of a shuttling that's happening within the cell organelle while the metabolite gets distributed. And this is not only in the rice, we have now done it in chickpea, this whole seed profiling. We also have this data for potato and tomato, and these are all unique and these are all generalized distribution that we see for all primary metabolites as well as the secondary metabolites. Which came up data, and as you could see, there are many of the loci that we found. Rice has 12 chromosome. So the most important chromosome in terms of this metabolite profiling turned out to be the chromosome 3, followed by chromosome 1, as many of the loci are harbored on these two particular chromosome. When we tried to do a pathway impact, we could find, as you could see here, that branch-chain amino acid metabolism, phenylpropanoid metabolism, aliphatic acid metabolism, metabolism in rice seed. And this shows you that TCA cycle is actually the major hub point which actually is related to any kind of this metabolism. As you know that TCA cycle comes from our respiratory pathway, and that is producing all the acids as well as many of the 1 carbon and 2 carbon molecules, which ultimately leads to the formation of the other secondary metabolites as well as primary metabolites. So once we understood that, then we tried to interrogate our data by pathway enrichment and to do a correlation of these rice metabolites with different other things. And as you could see, this positively regulated metabolites turned out to be in a red patch, where the negatively regulated metabolites, correlated metabolites, have been marked with something like blue color shade, and where you see only the cross, that means there is no positive or neither positive nor negative correlation that we see between all these kind of metabolite connection. As our data showed that the major pathways were carbon metabolism, amino acid metabolism, glycolysis, glyoxylate pathway, fructose, flavonoid, fatty acid, inositol, starch, and citric acid and pentose phosphate pathway. Whereas the minor pathways which were related to the metabolomics, that turned out to be the protein processing, amino sugar and nucleotide sugar metabolism, RNA transport, RNA degradation, and proteasomal pathway, along with the purine and metabolism and endocytosis. From this understanding that we understood from the IR64 data, then we now constituted a whole rice diversity panel for the nutrient analysis, which actually is coming from all the states of India. And we have taken a 12,000 rice genotypes across Indian agroclimatic zone. And from here, in our whole data set, we have landrace, wild relative, elite variety, mini core, ED3K panel, antenna panel, japonica, and all of those. And with this, what now we have been doing, we have been measuring, we have already completed more than 7,500 germplasm protein analysis, as well as more than 125 metabolomics analysis, 125 for QTL analysis. So now from here, when we try to understand all these nutritional component, the one part that is of today's aspect that the meeting has been organized, which is a post-harvest quality. So I'm touching upon one such post-harvest quality, which has been also published in this food chemistry paper, which is aroma. Now, this aroma, all these years, people know that whether it is rice or it is any other fruit crop controlled by one major metabolite, that is 2-AP. However, we understand that within the cell, it's not only one molecule gives one trait. We always know it's a multi-trait, right? That's why we call it QTL. And aroma is influenced by multitude of biomolecules that primarily regulate sensorial attributes actually in plant. And therefore, the aroma formation is not that simple. It's a complex process which is governed by what we believe by a synchronized developmental event and a biomolecular dynamics and a crosstalk of many enzymes and biochemical reactions. And there will be many feedback loop and regulation and a signal transduction. So to understand this whole pathway in holistic manner, which have never been done, and this was the first time we are showing this, what we did the work earlier in IR64. Now we included Pusa Basmati one, which is the most important aromatic rice that India is proud of, and came from IRI, from Dr. Ike Singh's work, and before that Dr. VP Singh's work, and that is Pusa Basmati 1. So we provide the same thing of the proteomics, of the genomics, of the metabolomics on Pusa Basmati 1. Then we try to compare one non-aromatic, that is IR64, versus one aromatic, which is PB1. And in that case, as you could see, how the water content and is related to the seed weight, and how it is different or similar with the IR64 and PB1. When we did that, we also measured the anthocyanin, and we also did the phenol and tannin, because as I said, these are something, anthocyanin is beneficial, but tannin and phenol are anti-nutritional property. We also did a scavenging activity assay, and as you could see from here, we found 406 metabolites which were commonly found between PB1 and IR64. However, PB1 showed 400 metabolite and IR64 showed 286 metabolites, which were very, very species specific. Now, from here, geoterm enrichment data showed again that amino acid metabolism, nutrient assimilation, carbohydrate, fat, storage, biogenesis, and signaling were the major component related to aroma responsiveness. What we then did is that we tried to make a regulatory network that is involved. Now, in this case, it is the regulatory network is only for the aromatic aroma formation trait, no other trait. So these are the proteins that are related to the aroma formation. So therefore, it is not only the 2-AP, but there has to be many more other metabolites. So what we did, this bad H2, which is actually the marker enzyme of 2-AP, when we measured, as you could, and very differently in case of 64 and PB1. However, if you see PDH and proline and many other things, this PDH and OAT and P5CS are actually needed for many other activity. Therefore, their difference is not that drastic. But if you look into proline and 5-PC, which are also related, and putrescine, which are also related to aroma formation, they show very differential regulation between the non-aromatic and the aromatic rise. Now, with this, what we did is we then actually tried to actually map the whole pathway and the biochemical events associated with aroma formation. And what we figured out is that out of all these things in between the aromatic and non-aromatic rise, what we found that the antioxidant response, transport, calcium and G protein signaling, and some of the transcriptional and post-translational pathways were very, very important. So in on top of the 2AP pathway. Now, along with this, what we also observed, and we did a MRM based quantitative metabolite quantification based on LCMSMS data, which showed the validation of this whole, whatever we are claiming, we got it validated quantitatively by quantifying flavonoids, cyclic polyol, and cyclohexane carboxylic acid. And what we then figured out is that in addition to this 2AP, which you could see signaling and amino acid metabolism, particularly branch chain amino acid metabolism, are also very much linked to the aroma formation in rice, and they will not be very different if you now look into aroma formation in any of the fruit crops or any of the vegetable crop. And we are trying to mapping all these things in tomato right now, and we would be possibly getting a chance. We are doing it in tomato and spinach, possibly getting a chance maybe in the next time. I will try to do the justice. So now this is what we actually understood from here. Now what happened is that our study therefore identified components of calcium signaling, G protein signaling, and postulated that these signaling events and the crosstalk of this signaling with the GABA metabolism actually governed this rice aroma formation. Of this, role of calcium signaling was earlier known in fragrance formation of other plants, but not in rice. However, involvement of this G protein is very, very unique, and this is the first time that we have been reporting it. Now, from here, then we wanted to map that what are the common pathway that you see between the aromatic and the non-aromatic rice for this kind of a post-harvest trait, which is the aroma, that is carbon metabolism, biosynthesis of amino acid, all these, glyxylate pathway, glycolysis, which are very, very same in any crop species. Now, when you see the enriched pathway, you see in the PB1, it is that 2AP, GABA, aromatic and branch chain amino acid, G protein and calcium, oxylipin, which is very important, cinnamoic ester, trypsin biosynthesis, mevalonate pathway, alkane and ketone, and phenyl pyrophosphate pathway that is enriched in the PB1, and therefore these are actually governing the aroma trait, which is a post-harvest state. In contrary, when you look into the enriched pathway in IR64, it is amino sugar, it is pentose pathway, it is RNA degradation, it is proteasomal pathway, it is purine metabolism, but neither of this pathway that becomes the highly abundant pathway. So we postulated from our data that what it is doing in the protein processing, which is happening in the endoplasmic reticulum through the ribosomal protein involvement, and that is related to all these protein processing machinery. Whereas the second pathway, which is amino sugar nucleotide sugar metabolism, that is by the TC cycle, fructose mannose metabolism and pentose phosphate pathway. The other one is interesting enough, which is RNA transport, and that RNA transport also has some relation with ribose sugar metabolism and cytosine adenosine metabolism, which in turn goes back to the where I said that amino sugar metabolic pathway in IR64. So from here, I come to my last part of the talk, where I'm going to tell you about the protein nutrition which is another post-harvest trait that is very, very important, as I have shown you from my initial data. So this is a protein which we have been working for a long time because of this unfortunate thing happening in my house. I could not get time to put my potato data into it, but many of you have possibly read two of the PNS paper long back where we have done this work in potato. Why I am saying this? Because this today's talk is on vegetable crop. So we already have worked on the vegetable crop. Anybody interested can Google two PNS paper. You will get one in 2000, one in 2010. After seeing that, so this is amaranth, which is a very, very lesser known crop, which is just our previous speaker said that this is an opportunity crop, and this that the DGICR is telling today is the amaranthus, which is many of you have used Ramdana laddu, and that is this amaranth seed. Now this amaranth seed, long back what we found is having a albumin protein, and this is the structure of the protein, and this protein has two agglutinin domain. We have, these are the reference, one can take the reference. We also have a patent, nucleic acid research paper, and BVRC. So using this, what we found that this has a very high protein content, as well as it has amino acid, which is essential amino acid. Those are very rich in this amaranth albumin 1 protein. What we now made is a construct where we are targeting this protein with a seed specific promoter, and with this we have generated lines, because as I said to you, rice has only 7% protein, and if we have to have proper rice with a proper intake of, the total protein content our body need should be 1 gram protein per kg body weight, and to meet that you really require 100 gram rice. If you eat in a diet, you need somewhere around 12% protein in rice seed, whereas whichever rice we are consuming today has only 7%, 6 to 7%. So in order to do that, we are trying to we have made all these lines, and we have done 4 years trial. In that you see this are giving us lines where we now have a protein content up to highest line is 14% of protein, and in that this also give a harvest index increase, and therefore it's also a beneficial trait for the farmer. What you could see is that the correlation of all the different agricultural trait that we correlated, and it shows quite positive correlation. With that, we did a biochemical characterization of among the all agronomic trait, and as you could see, some of the antioxidant property enzyme APX, SOD, amylase and LOX activity that we have measured, and if you see in comparison to the non-transgenic, that is the IR64, you see in all other lines you find that they are all very much similar. So therefore we are not touching any other trait while we are changing the protein content. Now these are the traits that showed harvest index, protein increase, photosynthesis rate, homozygosity, and yield. And all of them are positively regulated and showing a better result. So with this, we then tried to map the whole proteome of this AMA1 OS, and as you could find out, we map all these QTLs onto the different 12 chromosome of rice. We also did the same thing of the protein expression dynamics, wild type versus OS AMA1, and you could see the major pathway comes to the carbohydrate metabolism and amino acid metabolism, followed by lipid. And now from there, we did a whole metabolomics again with the GCMS analysis, wherein you could find that how in AMA1 in 3 different stages, milky stage, mature stage, and the germinating stage, which are the pathway is destined to the protein and amino acid formation, and these are all the pathway network study that tells us that in as against IR64 in the AMA1, you see more of the fatty acid and lipid metabolism, amino acid metabolism, and the sugar metabolism that we are observing. The most importantly, lysine biosynthesis, as you could see over here, goes much higher in case of AMA1 rice, and which is the most important limiting amino acid in terms of the protein quality improvement that we know. So to summarize this part of the talk, what we understood from this overexpression study is from the biochemical pathway, the biomolecules, the physiological agronomic trait and grain quality trait, which also has a post-harvest index trait, that is post-harvest quality, along with the chalkiness and cooking time, kernel elongation ratio, and grain dimension, which all of them are very important as of the post harvest quality, that is a cooking quality, is concerned. So with this I would actually conclude my talk. I am in a hurry, I am sorry about it. I'm again apologize to all the audience because I have to rush. So with that I conclude. The students who have contributed over the years is Konika, who is now a postdoc. Pragya is a PhD student working on that rice mission program right now. uh, she is a new PhD student, and she is working not in rice, but she's doing the same thing in chickpea. I have Bhanu, who did all the. We have now come up with the whole. Maybe we'll be putting a paper now where we have mapped almost 5,000 metabolite of rice, and that we are making a database. So Bhanu is helping in that. Atri is working on the rice nutrition. Pooja is a faculty now, and now she contributed a lot in the AMA1 rice work. Arunima also worked, and this is my current group. The whole work is funded by this part that I have talked to you, funded entirely by DBT and from a mission mode program, and some part has been actually funded by SCRB. And these are my current collaborators with whom I have been working on the Rice Mission program. So with that, I would be stopping here, and I would be happy to take any of the questions.
Thank you, ma'am, for your presentation. There are few questions from the audience.
Yeah.
Uh, Shubham, Shubham is asking, how does metabolite localization in different cell organelles affect seed development?
Just a moment. I can't see. Can I close my presentation?
Yes.
Then possibly I can see. I can't see the audience actually. That's it.
Ma'am, I'm just reading out the questions from the chat box.
Just okay. Now I'm back, so I can see all of you. Yeah, Shubham. So what was the question? That how the
Metabolite localization. How does metabolite localization in different cell organelles affect seed development?
Yeah, so it's a very, very interesting question. So it's not that all the metabolite will be which are differentially localized or dually localized in different organelle will be actually important for the development purpose. But yes, some of the metabolites which are commonly functioning both in the development as well as the nutrition, so they would only be important for those. So in that sense, if you think about carbohydrate metabolism will be one pathway which is very important in that. So some of the carbohydrate metabolism pathway biomolecules or the metabolite are dually localized, but they are important both for the development as well as for the nutritive value.
Okay, ma'am. Shubham, I hope you got the answer. You got your answer. One question is from Rajkumari. What is the significance of amino acid metabolism in developing seeds?
Significance of amino acid metabolism, it's very, very significant because let me tell you, I could not show you that slide. If you remember how seed actually develops, it develops from we have a zygote. From that zygote you have a seed development. Now when it is developing, in that case what happens is, in case of monocot versus dicot, you see two cotyledon versus one cotyledon. So in rice, when I'm talking, it is the endosperm where you have the most of the nutrient which are localized. Now you have actually, if you now see the seed development, you have a embryonic seed, you have a then heart shaped, torpedo shaped, then the mature shaped seed, and the milky stage to the mature seed, and then germinating seed. What happens during the nutrient, initially when the seed is developing, all the nutrient are synthesized in the leaf. From a translocation, all the nutrient are translocated from the leaf to the developing seed. Then it is a in vivo synthesis of the nutrient that happens because you get a detachment from the leaf to the mature. This happens even in the tomato fruit that we have seen. So in that case, within the seed, during this seed development stage, we call it as nutrient synthesis, de novo synthesis, nutrient accumulation. So heart shape, torpedo shape, those are de novo synthesis stage. When you come to the mature stage, it is the nutrient accumulation stage. And then when you see germinating seedling, then it is nutrient utilization stage. So there is a correlation between the seed developmental stages along with how do you see the nutrient dynamics into that. That's why I started by saying nutritional dynamics.
Yes, ma'am. There is one more question from Dr. Arpita, ma'am. Does
So
brand
no. So I did not fully answer the question. So the question was how amino acid synthesis. So the amino acid de novo synthesis is happening during that nutrient acquisition, and then those amino acids are getting fixed within the protein, and that is happening during the mature seed stage. And then you are getting all the storage protein that is coming, which is albumin, globulin, glutenin, prolamine. And then when you go to the germinating stage, those are getting again degraded, and then you see the carbon and nitrogen is coming out, and that's how again the amino acids are released. But that
I think there's some signal or
Yes, ma'am.
internet connectivity issue.
Yes, ma'am.
Shubhra, there was some connectivity issue in it.
now I'm audible, I think.
Yes,
Am I
ma'am.
audible?
Yes, ma'am. You're audible.
Next question.
Ma'am, next question is from Dr. Arpita. Does bran portion of rice contains more aroma components?
More?
Aroma components. Aroma.
Uh, Aroma in brand we have not measured, so I don't want to comment on that. We have measured protein, so I have commented on that. But what is known, I won't think that in the brand you will get more of aroma. Possibly not. As far as
I agree with you. Agree with you.
right, Nutan,
It's
if I'm
more
not
of
mistaken.
it's
So
more
we
for
haven't done it. Aroma bran is more rich into the protein and those kind of primary metabolic pathway, not the secondary
because
metabolic
the
pathway.
function of the brand is not to
No,
give
no,
the flavor. It's not the
no,
flavor is not the function of the brand.
no. It's actually for the carbohydrate mostly
Yeah,
and some
yes,
protein.
not for the protein.
Yeah.
Okay, ma'am. One last question is how does host pathogen co-evolution influence disease burden globally?
So this is another talk actually. Anyway, briefly.
But you have to summarize in 1 second.
Yeah. So no, briefly. Yeah, briefly it is that it's a One Health issue. I said to you. So Nutan will give me another chance. I come and speak on that in a 1 hour talk. So in that case, what happens is that coevolution is always there. So what happens is the pathogens are always evolving because it is a fight between the host and the pathogen, right? So therefore, who wins the race? That's the whole ball game. Now the pathogen are making different pathotypes, and similarly in the host our genes are getting evolved, and therefore there is always a battle between the host and the pathogen. So it's very important that we do not get more of new pathotypes, but it's not in our hand. Global climate is changing such that we would be actually expecting more of newer pathotypes which will be coming. You know that it says Sarah Good, who actually heads the whole UN, she says that possibly in next few years we now actually there was a time 1950s 60s 70s we were seeing more of communicable diseases which are pathogen infection, but then now we are saying NCD, that is non-communicable disease. But now again in next 10 years we'll be shifting to the communicable disease. That is the prediction because warmer the climate, heat is more, pathotypes are coming more, so we will be getting more of disease burden in next 10 years time. And therefore we have to be very very cautious for this multi-host pathogenicity that I have showed to you. You read a 2018 science paper, you would see how in the animal infection as well as in the plant infection, how in agriculture and clinic, and more importantly the fungal pathogen, because we don't have fungicide. All fungicides are coming from one molecule, that is, azole derivative, and that therefore these pathotypes are now immune to this azole derivative. So in front of us, if we can come up with a more other chemical molecule which can be a fungicide, that one should target right now to save us from newer pathotypes.
The mode of mechanism should be, the target should be the different one now. Thank you,
Sorry,
Shubha.
Nutan?
So the target biosynthetic pathway for fungicidal development
Yeah,
should be
that's true.
different.
That's
But
true.
a
That's
new
true.
target. We need a new target. Yeah.
That's true. It's very important. If someone I know in Amity now, I'm forgetting his name, who has joined, who is a very
Munish.
good biochemist, I think from.
Munish.
Mm,
Oh.
yeah, so should look into this aspect. This is very, very important aspect.
Yes. Thank you so much, Dr. Shubhra. It was such a wonderful talk. I wish we had more time so we
Yeah,
could
thank
listen
you,
to you
Nutan.
more. But we are
Sorry,
also sorry for your loss, and we are so thankful that despite that you remain committed for your commitment, and that shows how.
No, last time I could not make my commitment to this time. I said I have to go because last time also something happened, Nutan. And if you remember, you called me and I could not come. So I was feeling very bad about it.
No, it's not.
So I hope the
These
audience.
things are not in our control,
No,
actually.
that's not. But I hope the audience got benefited. If not,
Yes, it's very well. Like, I think the question and answer which we got shows that audience is very much enjoyed your talk, and they really have got new food for to think more.
otherwise anyone can contact me anytime.
Thank you so much, Dr. Shubhra. Now, Dr. Anamika, you can
Thank you,
uh,
Dr.
close it,
Anamika and
the session.
Nutan and whomsoever I could read out on my computer, Priyank Sharma and I don't know, Nishant, probably all from Amity.
Yeah. All the panelists are from MIT. Dr. Tandon has also joined. Welcome, Dr. Tandon.
Thank you, Dr. Nutan. Thank you,
Dr.
Ji.
Tandon is our additional pro vice chancellor.
Okay.
Dr. Shubhra is was former
Namaskar,
director of
Dr.
NIPGR.
Chakraborty. I just listened to your deliberation. Very interesting and very enlightening for everyone present there, including me. Thank you for being here with us. Really appreciate that.
Actually, she had some mishap at the family. Despite that, she remained committed for this presentation.
Oh, we are so thankful to you, and we pray to God that everything is resolved as soon as possible.
Actually, my mother is still, it's not a platform to talk, but mother, she was my mother's elder sister, and my mother is 88 years. So that's why I'm rushing back, actually.
Thank you so much.
Please. Thank you.
Please. Thank you so much. Thank you.
Thank you for sharing your valuable yes, ma'am. Thank you for sharing your valuable insights, ma'am. Before proceeding forward to valedictory session, I would request participants that we have shared a feedback form link and an assessment form link for current session and in the chat box. So please, you are requested to please go through the form and participate in the assessment as well as provide your valuable feedback for this particular session, session 5. So, good afternoon to all respected dignitaries, resource persons, and dear participants. As we come to the valedictory session of this FDP on emerging trends and innovations in post-harvest technology and management towards sustainable food systems, it is a moment of reflection, gratitude, and renewed commitment. Over the past few days, we have explored transformative ideas ranging from smart post-harvest handling, value addition, cold chain innovation, and digital supply chain solutions to sustainable waste management and circular bioeconomy approaches. These discussions are not merely academic, they are critical to reducing post harvest losses, enhancing farmers' income, strengthening food security, and building resilient food systems. We have also explored how advances in breeding strategies, biotechnology, trait selection, and integrated crop management can significantly reduce post harvest losses while enhancing the nutritional quality of the horticultural produce. These efforts are not only scientific achievements but essential steps towards strengthening food and nutritional security. In a world striving to meet the goals of Food and Agricultural Organization and aligned with the vision of United Nations Sustainable Development Goals, post-harvest technology play a decisive role in ensuring that what is produced with effort and resources actually reaches the customer efficiently, safely, and sustainably. I sincerely thank our eminent speakers for sharing their expertise and insights, and all participants for their active engagement and thoughtful contributions. Let us carry forward the knowledge gained here into our classrooms, laboratories, research projects, and extension activities. May this FDP serve not as a conclusion but as a catalyst for innovation, collaboration, and impactful action towards sustainable food systems. Thank you, and I wish you all great success in your future endeavor. I now invite our Dr. General Ma'am, Dr. Nutan Kaushik Ma'am, to give her remarks and overall summary of the successful completion of the 5-day FDP program. Thank you, ma'am. Over to you.
Thank you, Dr. Ramika. Very good afternoon to all of you. On behalf of our Honorable Founder President Sir and Chancellor Sir and Vice Chancellor Madam, I'm welcome you to this valedictory session. Every good thing has to come to an end. I would say not end, but and this is not the end of any program, but this is the beginning of new knowledge era, where whatever knowledge you have gained, you are going to take it forward. So, the primary objective of this 5-day faculty development program was to strengthen academic and research capacity in the field of post-harvest technology and management. With a clear focus on developing sustainable, resilient, and technology-driven food system. The program aimed to equip faculty members and researchers with advanced knowledge in smart post-harvest handling, waste minimization and valorization, digital transformation of supply chains, emerging storage and packaging technologies, and crop improvement strategies to reduce post-harvest losses and enhance nutritional quality. It is also aimed to promote interdisciplinary learning, foster innovation-driven research, and create a platform for meaningful interaction between academia, industry, and international experts. I am pleased to share that these objectives were successfully achieved through the participation of more than 100 faculty members and researchers daily, and though the delivery of 5-day keynote lectures and more than 15 uh, invited expert talk. We had 5 keynote lectures and around 20 invited talks and from by eminent scientists and global experts. So the outstanding response, active engagement, intellectually enriching discussion throughout the program clearly demonstrate that the activity has significantly enhanced knowledge, strengthened academic competencies, and inspired participants to adopt the emerging technologies and sustainable approach in their teaching and research. On day one, the theme focused on smart post-harvest handling and physiological management of fresh produce. The keynote lecture by Professor Ram Astre on smart post-harvest handling and post-harvest management established a strong scientific foundation by highlighting advanced physiological and technological interventions to maintain quality and extend shelf life. This was followed by expert lecture by Professor Dheeraj, Dr. Rahul Kumar Anurag, who delivered an insightful talk on modified atmospheric packaging of fruits and vegetables. Dr. Vasudeva Kiyar also elaborated on scientific approaches for physiological management of fresh produce. These sessions successfully enhanced participant understanding of post-harvest physiology, packaging technologies, and quality intervention. On the day two was focused on waste minimization, valorization, and circular economy, and the keynote was delivered by Professor Ajit Mishra, and which titled "Waste Minimization, Valorization, and Circular Bioeconomic Approaches, Sustainable Food System, and Upcycled Foods." This session provided deep insights into sustainable food systems, highlighting green processing technology, precision agriculture, innovative strategies for converting agricultural waste and food waste into valuable products. The invited lecture by Dr. Charanjit Kaur emphasized on value addition, particularly focusing on the pectin, how the pectin can be used as a bioactive compound from processing waste. Whereas Dr. Ashutosh Upadhyay had highlighted sustainable preservation technologies, and food safety interventions. Dr. Jasudeshri provided valuable insights into sustainable post-harvest management and resource optimization. Dr. Gaurav emphasized how the microbial resources can be used in the waste from the food industry can be used for as a food for the microbes for producing very highly valuable compounds. Similarly, in the day 3, Dr. Dushyant Taki talked about what are the new innovations which are happening in the field of the supply chain management, particularly for the perishable products like the fruits and vegetables. And similarly, Dr. Sri Ravi Shankar Sharma also highlighted the digital harvest, and he gave various examples of the various apps being developed. Professor Giancarlo Colelli from Italy, he gave very successful examples on post-harvest technology developed by his institute and the quality and increased market value. Dr. Maria, she gave a very successful example of the supply chain from the Dell and how the same supply chain principles which were applied by the Dell can be applied in the post-harvest technology. In the day 4, we was focused on the storage and packaging basically, and the keynote by Dr. Rajeshwar Mache, he gave a lot of success stories in the case of the packaging, and he also highlighted that what the biodegradability means. Not all bioplastics we developed are biodegradable. He emphasized the LCA approach rather than just the biodegradability as a marker for the sustainability. Similarly, Dr. Ajay Yadav and Dr. Shruti Shukla, they also gave various examples in the packaging from edible film to the other examples. Dr. Nagendra and they in the day 4, we also had the keynote of the day 5, Dr. N.K. Singh, because he had to he was not available today, so he is preferred to give his presentation on day 4, and he gave various approaches, molecular breeding approaches, which can be applied for the post-harvest loss minimization, including the shelf life enhancement and the delayed ripening. Today's session was on crop improvement for minimizing the post-harvest losses, and we had very eminent speakers, Dr. Shashi Bhushan Tripathi, Dr. Jagesh Tiwari, and Dr. Shubhra Chakravarti. All are leading experts in their field, and they all successfully demonstrated how this genomic technology can be used in the post-harvest minimization, and each one of them gave a very elaborative examples from their own experimentation from their own research which they have done. So likewise, we have, I think all of you have been gained immensely by these presentations which we could gather, and I would just like to give you some PPT, small PPT, giving you the glimpses of this 5-day FDP. I'll just share my screen and that provide you the glimpses. This has been compiled by my colleagues, so I'm thankful to my colleagues for quickly putting up all these things together, all the session coordinators, the convener and the co-convener. So these are the, so if I just highlight what the, the innovative ideas proposed by each speaker and the future research which is pursued. So Dr. Ramasri, he's talked about the scaling low-cost storage technologies, long-term safety, efficacy studies, and 1MCP integration of the farm technology. Dr. Neeraj gave an idea about the AI model for the real-time quality prediction, small logistics, traceability platform, academia, industry collaborative research. Similarly, Dr. Rahul Kumar Anurag, he gave very crop-specific optimization at microbial culture advancement, advanced edible coating, such and sensor-based atmospheric monitoring. Dr. Basudeva, he gave an integrated value chain research model, policy-linked post-harvest innovations, and institutional role in farmer-centric solutions. And these are some of the overall feedback on the session 1. So we, I think 86% respondent gave the speakers as excellent. And this is from the session 2. And from the session 2, we had the Dr. H.N. Mishra, and the future research which he proposed is scale up of the upcycled food, LCA studies, EI supply chain, techno-economic feasibility of biofuels. Dr. Charanjit Kaur, she emphasized on green extraction optimization, stability studies, industrial validation, and functional prototypes. Dr. Ashutosh Upadhyay, he proposed to focus on bio-based antimicrobial packaging, predictive microbial modeling, and regulatory studies. And Dr. Suresh, she talked about the low-cost storage, smart packaging, and climate-resilient technology. And Gaurav Rajoria, the biorefinery scaled-up fermentation optimization and clinical validation and commercialization pathways. And this, the 74% respondent mark the speakers as excellent speakers, the 13% as a very good speaker. Is the speaker quality. And then coming to the session 3, Dr. Dushyant Tyagi, he gave the future proposed research, AI-enabled marketing, analytics, digital inclusion of smallholders. Mr. Ravi Shankar, he gave an idea for the predictive shelf life modeling and integrated data platform. Dr. Kolili, he suggested to focus on the sustainable storage system, non-chemical preservation. Dr. Maria focused on the gave that focus. Our future research focus can be on resilient digital ecosystem, global SCM sub collaborations. And in this session, 81% respondent said the speakers were excellent, and with 22 having the very good, and overall rating of more than 4.75. And then the session 4, emerging technology for storage, packaging, and shelf life extraction. The Rajeshwar Mache, he gave the future research focus, should focus on high barrier biodegradable biofilms with scalable smart integration. Dr. Ajay, he proposed the cost-effective intelligent map for export supply chains. Dr. Shruti Shukla, she gave that future research can focus on scalable nanomaterials, validated mitigation safety. And then Dr. Nagendra Kumar Singh, he said the integrated genomic phenomics with bioinformatics infrastructures. And in this session, 57% respondent marked the, 80.3% respondent marked this as excellent and 12% as very good. And these are some of the glimpses of the FDP, some of the screenshots. I think you will enjoy them. So this thing, all the sessions, and thank you so much. I hope you enjoyed the FDP. Thank you for joining. And I think at the end of the FDP, we will be giving you the feedback form, and we will request each one of you to fill up the feedback form. So over to you, Dr. Anamika.
Thank you, ma'am. Now I would request Professor Dr. Chandradeep Tandon sir, Additional Pro Vice Chancellor and Dean Faculty of Biosciences and Biotechnology, Amity University, Uttar Pradesh, to please share his insights.
Thank you so much. I am audible. I hope Dr. Nutanji.
Yes,
sir.
we are audible.
On the behalf of our most respected Honorable Founder President Sir, Honorable Chancellor Sir, respected Vice Chancellor Madam, on my own behalf and on behalf of Dr. Nutanji, I once again welcome everyone for this valedictory function, which has been concluded very fruitfully, I would say. Of course, fruit we are still to see in the long run. Having said that, this 5-day faculty development program, I guess, has really thrown light on the emerging trends and innovations in post-harvest technology and management towards sustainable food systems. As I could see, Nutanji has very nicely summarized this 5-day development program and all the activities and the events under this. Starting from specifically, part which is really interesting is the academic rigor and the certification model and the daily online assessment conducted after each session. And we are now looking forward to outcome-based FDP model aligned with measurable learning outcomes. It was a pleasure to listen to a few of the speakers, especially I just listened to Dr. Shubhra Chakrabarti and others also. And representation from all the institutes, with ICAR, IIT, CSIR, Central Agriculture Universities, and also international universities, industry leaders and global research organizations is really appreciated, and this will surely lead to a strong academia, industry, international integration that is need of the hour. So now the point for us is now to take it forward so that all of us benefit from this, including each and every participant who attended this online session. I would also request Dr. Nutan Kaushik ji that maybe same workshop could be conducted next time in an offline manner, perhaps if it's possible, Dr. Nutan. So that will definitely, add
Yes,
to
sir. Yes, sir.
add to the value and everything. Though definitely this is a very I would say great move on your part and great initiative which you have taken. So having said that, I once again congratulate all the organizers, especially Dr. Nutan Kaushik and the team, whole team. I can see Abhishek Verma and others who are here, Dr. Sachin, there's so many people, Dr. Anamika, Dr. Nishant. So all our colleagues who have participated and who have been behind, sometimes, there are people who keep on working very hard, but they're not so visible. So those people actually play a pivotal role also along with the organizers. Thanks to everyone once again. Thank you so much. I really appreciate it. Thank you.
Thank you, sir. Now, I would request Dr. Nishant Kumar, sir, Assistant Professor, AFAF, as convener of this FDP, to please give vote of thanks.
Thank you, Dr. Anamika, ma'am. Very good afternoon to all the respected dignitaries, distinguished guests, dear participants. It has been such an honor to be part of this magnificent event. On behalf of the MIT Institute of Horticulture Studies and Research and MIT Food and Agriculture Foundation and the organizing team, I would like to extend a warm vote of thanks at the end of our 5 days faculty development program on emerging trends and innovation in post-harvest technology and management towards sustainable food system. During the FDP lecture sessions, the experts emphasized the recent advances and challenges in post-harvest technology, processing, management, waste valorization towards the sustainable food system. This event would not have been possible without the guidance, insight, and active participations of many eminent leaders, scholars, and experts. First and foremost, we would like to express our profound gratitude to our Honorable Founder President, Dr. Ashok Kumar Chauhan, for his unwavering vision to providing a world-class platform to fostering academic excellence and research innovations. His visionary leadership and commitment to nation building through education have been the foundation of MIT's success. Our heartfelt thanks to Honorable Chancellor AUP and President RBEF Dr. Atul Chauhan for his guidance and support to encouraging a culture of research and development to aligning with the global standards. We are also grateful to Professor Balvinder Shukla, Vice Chancellor of MIT University Noida, whose unwavering support has fostering academic excellence and innovation. Our heartfelt thanks to Dr. W. Silvamurthy, President of the MIT Science, Technology and Innovation Foundation and Chancellor of MIT University Chhattisgarh for his guidance and research and innovation. We would also like express our gratitude to Professor Charandeep Tandon Singh, Pro Vice Chancellor and Dean of the Faculty of Bioscience and Biotechnology, for his guidance and supports. Our heartfelt thanks go to Professor Nutan Kaushik ma'am, Director General of MIT Food and Agriculture Foundation, Head of the Institute of AIHSR, for her tireless efforts in organizing this workshop to addressing the challenges in post-harvest management, technology, value addition to enhancing the food safety, security, and the circular bioeconomy. We would like to express sincere appreciations to our keynote speakers and invited speakers to share their insight and valuable knowledge in their respective areas. We are privileged to have learned about the innovations and advancement in post-harvest technology management, their integration with the nanotechnology, artificial intelligence, packaging, value addition, and circular bioeconomy by waste valorization from our distinguished experts. Beyond our experts and speakers, the success of this FTP belongs to many. I would like to also commend Dr. J.S. Sodi, Senior Vice President, IT Department, MIT University, Noida, and his team for their coordination, technical support provided throughout the FTP program. A very special note of gratitude to Professor Nutan Kaushik, Director General, MIT Food and Agriculture Foundation, and the team for their diligent work to planning, guidance, and organizing his this faculty development program. I would like to thanks convener of this faculty development program, Dr. Abhishek Sharma, Assistant Professor AFAF, and all the session coordinators, Dr. Gautami Sivasmi, Dr. Devi, Dr. Priyank Sharma, Dr. Sachin, Dr. Anamika Verma, Assistant Professor AIHSR. Finally, I would like to thank all the participants joining these 5 days online faculty development program from India and foreign countries. In conclusion, the faculty development program has provided us with the rich perspective on the emerging trends, innovative technologies in post-harvest technology, management, sustainable food system, circular bioeconomy. So we trust that these ideas and collaborations sparks over here will bear fruit in future research and collaborations. So once again, on behalf of the AIHSR and AFAF organizers and the entire team, thank you so much to each of you for your valuable contribution. We look forward to your continued support as we strive together for a sustainable food system by creating innovation in post-harvest technology management, valorization, value addition, and circular bioeconomy. So thank you so much.
Thank you, Nishant.
Thank you, ma'am.
Thank you, Nishant. Good. Good job. So finally, we are Dr. Anamika, any link for the FDP? Dr. Nishant, for the feedback link?
For session, I have shared for whole FDP. I think Abhishek sir and Nishant sir will share.
Ma'am, can I share link?
Yes, please.
Yes, in chat box.
So, dear participants, please fill. We have a separate link for the overall FDP feedback. So every day we had some feedback about the speakers and also the evaluation form. This particular link is for the evaluation of the entire FDP. So please fill this form. We will give you 15 minutes. So by 3:45 we will close this session. So you have 15 minutes to fill these forms. So I would also like to, if any one of you would like to share your experiences. Dr. Tandon is here still or not? If any one of you would like to share their experience, please raise your hand. I will put, and IT, please put the video and audio on for these 5 participants. I would like to have, if they can raise the hand. I would like to have the experiences sharing from the participants.
I think Surya Pratap Singh has raised a hand, ma'am.
Okay, Surya Pratap ji, IT Surya Pratap ji का mic and video on कर दीजिए please. हेलो पुनीत हुआ कुछ?
मैम कर दिया है वो इनको.
अच्छा, Surya Pratap ji, you can you switch on your video and audio.
Good afternoon, ma'am. Am I audible?
Yes, you are.
Very good afternoon to all of you. Especially very thank you, ma'am, Nutan Kaushik, ma'am. Actually, post-harvest management, it is my research area, and I have completed my PhD from Dr. Y.S. Parmar University. So I have done work on post-harvest management, and this FDP is really very helpful for me. And I have taken lots of ideas how to perform something to design a research related to the post-harvest management. For example, active packaging and intelligent packaging. And this session, almost all the session was wonderful. And especially first session, Dr. Ram Bharose sir taken. And second one, I think Charanjit Kaur ma'am also given wonderful session. And one session was taken from NIFTAM, Dr. Ashutosh Upadhyay sir. So almost all the session was wonderful, but these 3 sessions I would like to give a number 9, 10, and 9 almost. So Ram Asre sir, sessions was wonderful, and also Ashutosh sir also given a wonderful session. So my experience is I think I'll give a 10 out of 10, ma'am. So thank you so much, ma'am, again. And thank you so much, Priyank and Gautami, ma'am.
Hello. Hi, Surya. Thank you so much.
Okay, thank you, ma'am.
Thank you. Thank you. Anyone else? Anyone else would like to share the experiences?
Nishant sir, please share it to everyone. You are
Yes, sir.
sharing
I
to
did
host and panelist.
check.
Just click on the host and panelist. You will see the list.
Everyone. Click it to everyone. Everyone.
Yeah, I did.
Any other participant would like to share the experience? Please raise your hand. We'll.
I think Susmita ma'am, she wants
Okay. IT Dr. Sushmita को please mic and video.
मैम एक्सेप्ट करेंगे वो तभी आएंगे एक्सेप्ट कर
Mr. Omkar has also raised the name.
Okay. So, Dr. Sushmita, are you able to?
Yeah.
Good.
Yeah.
Good to see you.
Thank you, ma'am.
Great
to see you. And the whole 5 day was very exhaustive, I should say that it was throughout the FDP which has given means many different area to see through and go through. So since horticulture crops, I have gone for the in vitro, but this has given again the different aspect, different area of horticulture crops which we can excel with, relate to the biotech. And everything was throughout. Even just I missed one of the session for which I was regretting after reaching home. Also, I couldn't join that, so I missed that session too. So really, each and every session was very nice. All the speakers were at par, means expertise in their own field, and definitely we will be gaining many things from this FDP. And ma'am, your program is always to a different level. That is very means heart touching, and I really love all those programs where whatever you are doing.
Thank you, Susmita, for your love and affection. Thank you.
Thank you, and thank you to all the team members who were there. Gautami, Abhishek, Devi, Nishant, Priyank, and all the doctors who are there. They did very wonderful job. Thank you so much.
Thank you. Before you leave, Susmita, can I request all the panelists to switch on their cameras and put the screen? IT, can we have the tiles in the tiles mode so that we can take a screenshot of all the panelists and. Is it really possible? Or I have to do it at my end?
आप अपने एंड पे कर लीजिए गैलरी मोड
अच्छा ठीक है कर लिया गैलरी मोड कर लिया आई सी अभिषेक आई सी डॉक्टर देवी आई सी प्रियंका आई सी अनामिका आई सी मिस्टर ओम धमाल I see Surya Pratap and Dr. Susmita. So all 5 coordinators are here, co-convener, co-convener. Nishant, I don't see Nishant. Nishant? I don't see 1, 2, 3, 4, 5 conveners are here. Nishant, now it's a complete panel now. So I'll just take a screenshot. Okay, all of you can take the screenshot now, so that in case I miss something. Thank you. I have taken the screenshot now. Thank you, everyone.
Thank you, ma'am.
Thank you, ma'am. Thank you.
Thank you,
wonderful
everyone.
opportunity, ma'am. You have given a wonderful
So much, ma'am. Thank you so much.
Thank you. Thank you, everyone. Thank you.
Thank you, ma'am.
If everybody is done with the any announcement? Any announcement?
No, ma'am. The link only the 5-day session was there, not the overall. I think that was the final which has been given to in the chat box. Because it was announced.
Final FTP, overall FTP assessment.
Overall was not there, ma'am, in chat box.
Nishant,
No, ma'am, I just
We have
shared.
provided. We have provided.
I just
Just now we have
shared.
provided.
I think Mr. Omkar also want to say something. Please, Dr. Omkar ji, please.
Yes, very good afternoon, everyone. Am I audible?
Yes.
Yes. Actually, the overall session was very nice. And actually, I am new to the academics, so I also got to learn very, very new things from this FDP.
Okay. Where are you based down?
Hello.
Where are you based down?
I am in Vadodara, Gujarat.
Okay. Good. Very good. So, hope which session you enjoyed the most?
About post-harvest losses and temperatures.
And temperature. Storage.
Also, yes, because I was in Mahindra Agri Solutions Limited in 2022.
Okay.
So I was working with the same.
Okay.
I control the temperatures. What are the pre-cooling? How to do pre-cooling?
The storage and all that. Okay, good. Very good. Good. Post harvest storage. Good. Very good. Excellent. Good. Thank you so much for attending. Thank you. I hope you will apply some of the knowledge which you. We would be very happy if you can just share in future. We will be doing a survey 1 year after this FTP, asking each one of you if you could apply this research into your academics or in your research or somewhere. Okay?
Yes, I'm sure. Yes.
Okay, thank you so much.
I also my own research in dragon fruit wine also.
All those who have participated in this FDP are now the METL winners. Okay. Okay, thank you. Thank you so much. Thank you. Okay, anyone else?
Thank you.
Okay, thank you. Thank you, everyone.